Luke is going to kick us off with the intro to this session on sustained observations of global ocean biology.
Great, thank you, Heather. So yeah, this is our last session. We're kind of bookended by biology, which is great for the biologists in the crowd at least, hopefully for everybody too. So this session is called Sustained Observations of Global Ocean Biology, and I want to thank my co-chairs, Adam Martiny, Alyse Larkin, Zach Erickson, and Susanne Craig. So the question I want to leave you or lead with is, how do we know that the ocean is changing? This seems like an obvious question maybe, or the answer should be obvious, but let's take an example. So if we were measuring CO2, right? If we just had a few measurements like you see on the left, maybe CO2 goes up and then it goes down and then it goes up, you can't really make sense of it, right? Well, now that we have, people had the foresight, and now that we have 50, 60 years of measurements, we can see that there's a seasonal trend, but there's obviously a longer term trend that is more concerning. We also have similar measurements in the ocean. So some of the ways that we collect this kind of data are large cruise programs like Ghost Ship. And they're sustained, but unfortunately there's no biology on them, at least up until a few years ago. So there's all sorts of physical and chemical measurements that have been taken, but no biology. And that's kind of a trend across lots of different programs. There are some long-term sustained biological sampling programs, but not at the scale that's required to answer some of the big questions about our ocean. So the theme or the motivation for this workshop is that ocean biology is undersampled in time and space compared to physical and chemical properties, that there's uncertainty in ecosystem responses to changes, whether natural or anthropogenic, and that new technologies are making it possible to expand biological ocean observing capacity. And therefore, we're getting now biological data and knowledge. And so what have we learned? What are the challenges remaining, and where do we want to take this? And so just to give you a flavor of the kind of measurements we can take now, there's genomics and transcriptomics, flow cytometry, various optical measurements, flow cytobot, UVP, et cetera. So, and a lot of these are being deployed now on some of our ocean expeditions, such as Ghost Ship. And so today you'll hear about a few of those sampling programs. There's a lot more that you may not hear about, but there are some really nice programs, and they have been going on more than just the last few years, some of these, but the big oceanographic programs like Ghost Ship, and there's also the Marine Biodiversity Observation Network that you'll hear about, are some of the efforts that are really starting to add biological observations in a sustained way. And so just to give you a preview of the discussion we're going to have at the end, so you can start to think about some of the questions that we're going to be posing. Why do we need sustained observations of ocean biology? Is there an alternate viewpoint? Is there maybe a reason to not commit to sustained observations? Because of course it does come with a cost. How are current observing networks, sustained observing systems, and technologies helping us observe ocean biology? And what improvements are needed? What are the barriers? And how can new technologies help us accomplish sustained observing? How should we prioritize different observations? And what are the biggest challenges and biggest opportunities with regards to biological data management and data sharing? So of course, there's lots of data that's generated. That's wonderful, but we have to figure out how to deal with it. So with that, we'll kick it off. And let me pull up the agenda. Got it. All right. So looking forward to a good session. Thank you.
Good morning, everybody. I know how it is to be the talk after the banquet. So hopefully this will be a good story and one that sort of resets your brain. And I feel like the subtext of this could be, I'm telling you what you already know. So I'm Paula van Tempe. I'm at the Graduate School of Oceanography at the University of Rhode Island. And they asked me to give a talk that sort of sets the big picture stage for what we're going to talk about this morning. I think you're going to see a lot of great talks this morning that talk about new technology, new ideas, new approaches for making these observations. But let's take a little journey this morning, and hopefully this will be okay. All right, so observations. What are we trying to observe and why? So in the 18th century, there were a lot of whalers who knew, Folger was one of them, cousin of Ben Franklin, knew that the motion of the water that we now know as the Gulf Stream was headed in a certain direction, certain temperatures, certain biology was found there. And they just had their crossings to sort of identify what was happening there, no real formal sampling. And I often wonder if there was any point that Ben Franklin would know that a couple of centuries later, this is what we could see in the Gulf Stream, right? And this is a Modus Aqua image from 2003. It's 20 years old at this point. And you say, could they have ever envisioned that this is actually what the first optical depth of biology in the ocean looks like when we look at it from space? So we've come a long way in a couple hundred years since we first envisioned what the Gulf Stream might look like. The question is, do we really understand what's happening there? And are we ready for the next couple of centuries of our observations of the global ocean? And how does that scale down to the local regions that we want to actually look at and some of the challenges that we have to manage? So what is an observing system? Okay, and I like using this schematic of exports. This is just something from 2018 when we drew out what the exports program could or should look like. And the interesting thing here is you see ships, autonomous platforms, airborne and other satellite observations, and then you also see the components scientifically of what we're trying to observe in the exports program. And what was the driving question, right? What are the export pathways, sources, sinks, fluxes of carbon in the ocean? And what are the biological controls on them? Now that's an oversimplification, but we design the experiment accordingly. Well, sometimes the observations are already there, and the question is, what are we after scientifically? It goes in both directions. There's opportunistic science that can be done with existing observing systems to take advantage of that. But it takes careful planning and it takes time. And we're going to come back to this. So thank you for that perfect intro. That was great. Let's go back to the beginning, right? One data point, one point in time actually gave us a view of what is happening in our global Earth system, right, with global warming, with the rise in atmospheric greenhouse gases, particularly carbon dioxide. One data point. So this is arguably one of the most significant works of the 20th century. And it's really the first significant evidence that the climate was changing. And we ignored it for a really long time. Well, we didn't, but others still do, right? And I like to tell people when they question this, that if your cholesterol looked like this, you'd do something about it. If your weight looked like this, you'd do something about it, okay? And this is not something that should be pushed onto the individual. Believe me, we recycle everything we can under the sun at home. We compost everything. We rid our house of single-use plastics. I drive an electric vehicle. Do I have that moment where I feel good about my, contribution to what's going on in the world? Yes. It is not my problem to solve. It is the world's problem to solve, okay? So without some fundamental shifts in what we're doing, we are not going to address this. So what we do know from this time series is that atmospheric carbon dioxide has increased by more than 50% over the length of the time series. And that is incredible. 2013 was the first time that we passed 400 parts per million by volume of carbon dioxide in the atmosphere. This is a recent development, right? I'm, again, preaching to the choir and telling you what you know. So an investment in that time series is utterly critical, but we know now that we have to go broader than just one time series, okay? And it is the most unsexy thing to sell, believe me. But I guarantee you most of you in the audience wear a Fitbit, look at your Apple Watch, count your steps. And the reason you do that, the reason you go to the doctor to get a physical every year is to know what's going on in your organism, in your body, so you can take care of yourself. So we can also reconstruct climate and look at natural versus anthropogenic variability by looking at cores. Ocean drilling, extremely important, okay? Going to our poles and looking at ice cores, extremely important. They allow us to reconstruct the climate over time, over millions of years, okay? So we have ridiculous access to what I would call ancient air, right, by doing this, very important. And so the carbon dioxide levels, of course, have varied over many glacial, interglacial cycles. And if we look at the last million years, basically it's the atmospheric CO2 has stayed between 172 and 300 parts per million. It's really only, do I actually have a laser pointer? I don't know if I do I? Okay, so the atmospheric carbon dioxide really in the last part of the Industrial Revolution after 1750, and really in the last 100 years, like we just looked at, is where it's skyrocketed, right? And we know that to be largely anthropogenically driven. So these records tell us that atmospheric CO2 levels in climate go back way before the development of human civilization and our footprint on it. And so this is the 300 parts per million line, okay? And the last time that atmospheric CO2 was at 300 parts per million, the Titanic sank. And if you don't know what year that was 1912. So we know what's happening here. And if we did not have access to this type of information, we could not reconstruct climate and know about those impacts. So the near-term single-point time series plus our ability to reconstruct historical climate in some of our paleo records is utterly critical. And making sure we're planning for the future observations, also very important. So, big surprise that I was going to show this, right? So the last 10 years were some of the hottest on the global record, okay? And 2022 right in the middle. Of course, we haven't done 2023 yet. So Earth's temperature varies every year. How do we know this? Okay, we take measurements of our global Earth, total temperature, we look at the anomalies, and we look at those deltas, and we know the Earth is warming. Now, yesterday morning, do not ask me why I needed like a moment of inspiration in my life. And so what did I do? I was digging through the latest IPCC Working Group 2 report from last year. And I did something I never do, which is I went to the summary for policymakers. I never read the summary for policymakers. I just go to the report and look for what I'm looking for, right? And there was something there that really struck me. And that was that Earth's population is now well surpassed 8 billion people. And between 3.3 and 3.6 billion of those people are in direct, immediate impact for sea level rise, okay? That is 40 to 50% of the world's population, give or take. And the majority of those are low wealth groups, right? Historically marginalized communities that have to be moved. And so how do we protect our shorelines? In just the state of Rhode Island, it's 413 miles of coastline with 7,000 properties and businesses along that coastline. Economically, that's a huge impact. Not to mention, we can't let that happen to people. Okay, so what steps do we have to take in our shared governance of how we actually address this problem? Because flipping back to that whole cholesterol challenge, can you go and get a statin to take care of that? Yes. But that doesn't fix the problem. You still have clogged arteries. You still have to fix that. So that's the systemic challenge that we're looking at here, not just in biology, but in the globe. So how do we actually know that the last 10 years are some of the warmest on record? Well, we have global satellite observations and modeling that actually tells us that. And if you look at this animation over time, looking at the global surface temperature anomaly data beginning in the 1880s and marching forward, we see that that anomaly is growing, and particularly at the poles. So the red colors are the larger deltas in temperature, the warming, right? And where you freeze it right here, this is a 5-year average from 2018 to 2022, and it's normalized against a 30-year norm, 1951 to 1980. So this is where we're headed. Again, you're all nodding like, I know, we all know this. We're telling, okay, not everybody, this doesn't register with everyone out there, okay? So satellite data, very critical, modeling very critical in confirming this. What else is really important is the Argo data set. We actually have to take measurements in situ to confirm and validate what we're measuring on orbit, and that's very important. So This is an old plot as of like September 2021, but there were almost 4,000 floats. My guess is they've probably surpassed that by now, and we have lots of other tools as oceanographers that we can go out and use to sample. We take research vessels out, very important. The academic research fleet, some of the private vessels, philanthropic contributions, all very important in facilitating our mission. Not just taking data in field campaigns, but actually validating some of the global observations that are out there. And ocean warming is just dominating, right? This is where all the energy is going, and 93% of the excess heat that's coming in that you saw demonstrated in the previous slides is absorbed by the ocean. And the question becomes, what are the grand challenges? Perfect setup this morning in the intro that we're trying to address. How much heat can or will the ocean absorb in the future? What's the capacity? And we're going to talk about ecosystem impacts in a minute. And then will there be a tipping point, or will that slow down? So the first proof of concept in looking at global ocean biology was the Coastal Zone Color Scanner mission, launched in 1978 and lasted until 1986. And it was our first glimpse, our first wide-angle lens with which we viewed global, regional, and local pigment, total pigment in the ocean, proxies for phytoplankton biomass, so to speak. HPLC was something we used regularly as oceanographers, not at all created for the oceanographic research community, created for the medical industry, but applied to actually validate some of the observations that we would take at sea as to what we're seeing on orbit. And I think Luke gave the really nice intro, some of the newer tools that have come in, and I don't have time this morning to cover all of them. He had a nice snapshot of them. But for example, the IFCB, right? Early models of it, can we use this to actually get at more specifics associated with what's in situ? And I'm going to come back to this because I know you saw a talk on peace yesterday. So investment in these tools, investment in industry, and a pathway to commercialization for some of these tools is going to be absolutely critical for us being able to validate and understand not only that the ocean is changing in its ecosystems, but what direction, the sign of that change and what's happening. So one of the other things we were able to do after the CZCS mission is launch the CWIPS mission and then later MODIS. And we were able to do things like merge land and ocean biospheres, okay? We were able to detail changes of things like the 97-98 El Nino Southern Oscillation and their impacts in the ocean. We were able to watch the Earth breathe. And this is absolutely important. The time series, that uninterrupted time series with subsequent satellite missions, no matter what we're looking at, gives us that continued wide-angle lens with which we can actually view our ocean and our land, for that matter, right? The land-ocean interfaces, very important. If the ocean were just a static box, that would be one thing, but we have an Earth system, and we have to look at the ocean's role in the Earth system as well. So taking this one step further, this is a really old animation of the Echo2 Darwin model. And when this came on the scene from MIT, it was revolutionary. And we were looking at phytoplankton down to the group level. And you can see the key on the bottom, general groups, diatoms, flagellates, synechococcus, and prochlorococcus. And I gave this presentation, I used this in a presentation not that long ago, a couple of years ago, and somebody came up to me and said, I think your prochlorococcus is totally wrong in this animation. And I said, yes, great. You know what that means? That means our models are getting better at showing this, but showing the distributions of phytoplankton in the ocean, those ecological impacts, that response to climate change. But we know we have to get out and sample more. And how do we get out and sample down to the cellular level, the group level, and actually validate what we're putting in our models based on some of the satellite data? And this actually captures 71 groups in the animation. So we do this by having in situ programs that are utterly critical. And people gag at the cost of some of these, but they're minor in comparison to the economic impact of not knowing the global change that's going on, the geopolitical challenges that come with it. So Go BGC floats on the left-hand side, just a snapshot that I grabbed off the website earlier this week, and things like investments in optics and other biogeochemical properties on Argo floats. This is the SOCOM program snapshot on the right. Just show what's got optics, what doesn't, and the very hard to reach Southern Ocean. all right? Passive radiometric satellite data cannot see through clouds. So how do we get information from the really hard to reach areas on our global ocean? Okay, we do this by sampling in different ways, and maybe it's suborbital, maybe it's all in situ. So we have to keep investing in new sensors ultimately, and those observations to quantify and narrow, first identify the unknowns and then narrow the uncertainties associated with them. This is the very root of exploration, discovery, and research, and it all feeds into management and policy. And we may not make those decisions in this room, but we want to have policy relevant science. That's ultimately what it comes down to. So this is absolutely critical in funding these field programs, whether that is actually floats, drifters, the field sampling from the ships, et cetera. And I know it gets expensive. We're going to talk about that in a minute. So BioArgo has just been a critical investment at the end of the day in the efforts to measure global biogeochemical proxies and validate things from space. So I just threw this in there just for Adam's benefit. No, I'm just kidding. But programs like BioGoShip are being used to collect things like omics data sets that are giving us a lot of information about what's happening in the ocean and what organisms like Prochlorococcus have in their toolboxes to utilize different nutrients that are in the ocean, okay? So this was a great insight into omics scale analysis of nutrient stress in the ocean. Now the question becomes, how do we connect this to the upcoming satellite missions that are on deck like PACE? Okay, so it's like the genes to seascapes kind of thought process that we have to start thinking about. Do we have the tools to do that? Because we are laying the groundwork, but we need to make that broader investment. So the next 50 years of ocean science, bringing it all home, are we ready? The SWAMP mission is out there. It was launched in December. This is like all water all the time. I absolutely love this mission. So it measures the height of all water on the face of the planet, whether it's on the land, whether it's in the ocean, et cetera, okay? And the idea is that you can link all the scales of all the interfaces in the Earth system by looking at the water cycle. So it's relevant to ocean physics, hydrology, ocean biology, and biogeochemistry. They expect to have data available sometime this summer in the next month or two. So if you haven't taken a look at the SWAP mission, surface water ocean topography is what it stands for, it truly does bridge land, ocean, and atmosphere, okay? So again, interfaces. PACE, you saw the PACE talk yesterday, many of you, the Plankton Aerosol Cloud Ocean Ecosystem Mission. You know, we're going to extend key systematic observations of ocean biology and biogeochemistry, aerosols, clouds, and some of the radiative forcing, which are the largest unknowns in our global climate models at the moment. We're going to make new global measurements of ocean biology and biogeochemistry as well. Unprecedented resolution of things we only dreamed of observing with CZCS and back in that era. So when we go and design validation experiments, those will be open. Those will be the opportunity to test some new ideas, build some new sensors and sampling platforms. Lidar is really in the next era, and this is a bit of an eye chart, but I'm just here to tell you, you know, we used the Calliope data from the Calypso mission as a proof of concept. It was not designed for the ocean, and that was in 2006. ISAT-2 will fill some of those gaps, but it's not so good, and we don't see things except at night, which is problematic for actually looking at some of the biological processes that we know and love. We have a potential gap, and then NASA is working with other space agencies to think about what's next. Really, we need a blue LiDAR, something that is designed to penetrate the ocean and can get to things like ocean biology and ecology beyond the first optical depth and down to possibly 3 to get that vertical distribution of actually what's happening in the ocean. And so I just also throw this in there as a placeholder to remind you that ocean atmosphere dynamics are also very important. Ocean ecosystems are really only one part of an interdependent Earth system, and there are two-way interactions that we have to acknowledge. So coming off of the NAMES project, looking at the ocean atmosphere dynamics are important. Okay, so I can close with this one. How much is this worth? NASA's Terra and Aqua satellites, when they were built and launched, were about $1 billion in 1999. And everybody goes, oh my God, $1 billion. $1 billion is what the drill ship will cost, what the Antarctic research vessel will cost. That's a pretty small investment for 30, 40 years of operation, okay? Pace came in at about $853 million at its outset, and ESA in 2013 to build 7 Sentinel satellites was about 2.5 billion euro. How many people carry a Fitbit and Apple Watch? How many people look at their steps every day? You don't have to raise your hands, especially after the banquet, okay? So if I believe a quick Google search, on an Apple Watch, there were 7.6 million shipped in the first quarter of 2020 to the tune of $70 billion of annual revenue just in that quarter. In quarter 3 of FY22, Apple generated $40.7 billion from the sale of just iPhones. And if you want to go retro, sorry, Fitbit, US about 1.2 billion in 2020 with 10.6 million units sold. That's 31 million people per week using a Fitbit. Do you guys know what the #1 use of those devices is? Can you guess? What do we do with those? It's not directions, it's tracking fitness, which is exactly what the point of doing these observations is. That is what we are doing in the ocean and in our Earth system. So I'll stop there. Thank you, guys.
Thank you, Paula. We have a few minutes for questions if anyone has them. We'll also have a panel discussion at the end.
No, I have a question.
Okay. I thought I was going to get away, Adam.
So a lot of the satellite missions, right, these are optical measurements. It's a very abstract way of looking at ocean biology and biogeochemistry. So you have to somehow connect them with something that you observe in the ocean. So what are some of the critical measurements you think that we need to really connect the satellite missions with understanding ocean ecosystems?
What are some of the critical observations?
Yes.
Yeah. Okay. So, boy, there's so much information out there, but the ability to put different scales of data together and actually understand it, I think, is one of the biggest needs right now. Like, do we have the ability to handle the amount of data that are out there? Plus, that's sort of in my mind as a one step at the most fundamental level. To get at your question, the omics data, I think, are an important step in the direction of understanding the cellular level chemistry and how things respond to climate change. I think actually getting at, from a satellite perspective, species level data, if that is even possible. You know, I started out my career as a phytoplankton taxonomist, and you'd spend weeks, months, years trying to determine what was happening by looking at water samples and settling things out. And then, you know, here comes satellite data saying, oh, we can do this on a global scale. It's really easy. Look, you know, putting those scales of observations together as to the phytoplankton and even the plankton in the ocean, I think is going to be really important. More global surveys of things like fish and fisheries, and not just in MPAs, right, marine protected areas, but actually the global ocean. If you haven't read the latest IPCC, you know, there's this poleward migration of fish species about 50 kilometers every decade. That is an insta-geopolitical challenge, right? So how do we pull together the holistic ecosystem of the ocean, if that gets at your question, is sort of what I worry about on a daily basis.
Great talk. I love your analogy between the chlorophyll and the CO2, and also the revenue of the iPhone and Fitbit sales to the investment of NASA. So the question is, I think also a question to everybody. So this is a capitalism-driven society. How do we make people feel that, you know, the sales of the iPhone, Apple Watch benefit shareholders? How the invisible benefit, future benefit, and we make people feel that it's useful?
Yeah, it's a great question. I'm not an economist, but I can just give you my completely subjective opinion. As I said, you know, whether you know this or not, carbon footprint is an idea that the oil and gas industry came up with. And what it does is it puts the burden of what's happening on the consumer. Recycling is from the plastics industry, again, to push the burden onto the consumer, right? I'm not saying it's wrong. I'm saying we should be engaged in that. I drive my electric vehicle. It makes me very happy when electric vehicle isn't going to solve the problem. We have to incentivize. I'm a firm believer in the fact that, you know, if we incentivize equally, globally, okay, and we support low wealth groups that don't have the ability to transition to electric vehicles or non-single-use plastic and things like that, then it's going to be a problem. But industry is very clever, and renewable energy is a whole field that I think could explode if just the proper incentives were put in place. But industry is also going to have to work with government to make this work, okay? If we do not change, then if I have one more person say to me, oh my God, we didn't get any snow, and usually New England gets so much snow every winter, this is a trend that's going to continue, right? I mean, we can't prevent that. The Earth has been around a long time. It'll be around when we're not here. It may just not be the Earth with the resources that you want. So I think that partnership between industry and government internationally is going to have to happen. By the way, 42% of the world gets at least one protein source that's critical from the ocean, and that is rapidly changing. So with statistics like that, we've got to do something, and we've got to do it together.
All right, let's thank Paula. All right, it's a pleasure for me to introduce Julie Robidart from the National Oceanography Centre in the UK. I feel like it's a little bit full circle here, as I met Julie the first time 22 years ago, right here in Woods Hole. She's an expert on developing new technology to really improve our ability to observe ocean biology. And as a testament to that, McLean Research Lab was demonstrating one of her instruments at the reception a few days ago. So I'm very excited to see her talk today. Let's welcome Julie.
Yeah, so it's great to be back here giving a talk, and I love coming full circle to where I became really a microbiologist. This is a formative place for me, and I absolutely have a nostalgia about Woods Hole. So thanks for the conveners for inviting me to this. And I'm really excited to talk to you about some transformative technologies that currently exist and kind of where we're moving for measuring biology in the ocean. And biology in the ocean is incredibly complex, right? It goes from, well, you could call viruses biology, but it goes definitely from microbes all the way to whales. And so that means that in contrast to parameters that we know are a little more specific, we can actually, we have to come up with very different tools and measure biology in very different ways. So I love this image over here on the right. This is from Kelly Lance at Mbari, and it's meant to represent the ways that we can measure biology. There's microbes on the left there. There's zooplankton, and you've got your fish and all your larger organisms. And what this shows you is, you know, there's no single case solution. You actually have to measure microbes. You can measure metazoans in the same way with the same tools, but actually combining them and using them differently for different types of biology is actually a lot more powerful and accurate. So I wanted to mention here that DNA, I'm going to talk a lot about DNA, but nucleic acids, that's molecular biology. I'm going to refer to it as molecular. It's molecular is DNA, it's RNA, and it's proteins. And it has the ability to get from taxonomy to function. So you could actually say this organism it has a capacity of doing this function using DNA, or it's actively engaging in this function using RNA or proteins. So molecular biology in itself is very powerful, but it can also be used like a forensic investigation. So you could use the DNA that's left in the environment by microorganisms to understand who's been there and actually what species to the species level have actually existed in that community. So that one filter sample, you just got to filter the sample, preserve the sample, bring it back to the lab. You can actually look at the entire tree of life. So So it's a really powerful thing to just go out there and filter. Microbiologists, being microbiologists, actually are very well trained. They tend to be really good at optics. They're really good at specialists in highly trained in molecular biology and molecular tools. And we go to sea and filter water. We spend a lot of time filtering water. Let's stop. There are tools that do this now. Okay, thanks. But microbes are also really important for the base of the food web, right? We know that that microbes are changing over time and space. They're changing because of climate as well. So it's complex, but it is tangible. You can actually measure baselines, moving baselines over time, and understand how things are now and how things are evolving as, let's say, tropicalization occurs. Because they have this very fundamental role at the base of the food web in oceans, they also have a fundamental role in biogeochemical cycling of specific nutrients. But you end up having this complexity that's compounded onto this crucial role, right? And it means that we really need to get a handle on this, and we need to be doing things better and more comprehensively across marine environments to understand really how they're going to change over time. So the American Society of Microbiology put together kind of a global look of what we need to be doing, so research recommendations to understand how microbes are changing with the changing climate. And these are actually very similar to what we are saying in OCB. So we need to understand how metabolic fluxes alter as climate changes, because that's an ecosystem service they provide. We need to be more interoperable in our data so that we're not saying different things because of the types of data that we're interpreting. And then we need to incorporate greenhouse gas cycling, because we know that that's a crucial role that microbes are involved in. And then we need to assess innovations for their societal and scientific benefits. There are changing threats to coastal health, and I'll just say this briefly, that we know, we've heard a few times now, that eutrophication is going to lead to deoxygenation. These are not super predictable events at the moment, but we know that there's a causation there. In addition, ocean acidification is really making life hard for corals. They're declining, and bivalves as well. Physiologically, they're just not as capable as they used to be. And this is all compounded by industrial change. So we know, at least on a regional level, aquaculture is going to have a big impact in coastal regions. There's going to be applied research to do with aquaculture as well for, let's say, monitoring sea lice and things like that. But this eutrophication, this changing in the chemistry and the biology of the ocean is definitely increasing at the same time as offshore infrastructure is increasing. So offshore wind is going to kind of form these artificial reefs. There's going to be a change from the natural state of the ecosystem. So it's really a good time to start measuring more in coastal regions. And I think for biology in general, it's imperative that we start doing this now. There are a lot of ways to measure the environment for biology. So the marine environment on the left there, we need the physics and the chemistry, because the biology data just really, you can't scale it up. You can't do a lot without it if it's just the biology data. But in addition, you can use acoustics for mostly metazoans, or imaging, or optics and molecular biology. They have their benefits and their disadvantages and advantages, but molecular biology really give you an in-depth function and taxonomy, but the imaging techniques allow you to add on abundance and biomass, and really that link to historic data sets, because we know programs like the Continuous Plankton Recorder program has been measuring for decades using microscopy. But they both are amenable to faster collection. We can actually develop flow-through sensors and get data back in a quicker way using both these techniques. I'm going to focus on them because this is a microbiology. What does it have to do with carbon? We're going to talk about microbiology because metazoans have a more tenuous link. So across ocean gradients, we've actually heard about these. I'm not going to get into it because Nicole's going to talk about the BioGoship program, but I wanted to highlight on the left here. I like this this concept from Clayton et al. Of taking your scientific question and then using these approaches, and these are all imaging and omics approaches, to ask those fundamental questions. So a combination of these will be the right tools to answer the questions on the left. In addition, for imaging, I'm just going to mention imaging here, and then I'm going to move on to sequencing. But all these tools exist, and in the last decade, the automated classifiers have really blown up. So it means that the reach of these is a lot broader than it used to be, and that means the resolution is going to be higher and the impact much greater. This is a Terra Oceans expedition, but they combined this with DNA and physical and chemical data as well. So there we go. This is a crude, roadmap. We've got a more detailed roadmap for technology, but what I'm going to focus on today is observations and validation, linking that to biodiversity baselines and drivers, and that's really going to inform our models. So we're going to be able to say more about things globally if we have those things linked. The next place things are going are onto rates. A lot of people are asking for sensors for rates these days, and that'll have a similar impact with understanding drivers of flux and functional models. Across the way, so I should say, when I deploy something for the first time in a demonstration, I always do a data dive of the region, and I understand, what are the big questions here, and that really increases your impact with that first deployment, those first proof of concepts. And we should be doing more of that. The engineers need to be integrating a lot more with the scientists. But along the way, you generate more and more hypotheses, and that will stimulate a lot more process studies that can be independent. There's loads of technologies to scale eDNA and omics over space and depth. So I'm turning now to just DNA. This I wanted to highlight because this could be a workhorse for coastal regions. So there's a picture on the bottom left there of Michael, and he's got a sampler in the water. This is the accessionist pump, and it just filters down on a 45 millimeter filter. You could put this off a boat really easily. It's got seven channels for sampling. This is really nice for citizen science, for really scaling up what we're doing with colleagues that aren't traditionally omics people. On the left here, this is the Roxy. This is the robotic cartridge sampling instrument that is now commercialized as McLean, so you could buy one. And what it does is it has a Stereovex filter on a bandolier, which you can see on the top right picture, and it filters and preserves. So there's a filtering stage and then a preservation stage, and you could really just put a load of them. They could go on the bottom left there. That's Robin Samuel, and on the top left is Rob Brown, and they're small footprints, so they could go on moorings or they could be deployed in situ. I'll get into that a little bit more later. But this long-range AUV has been now integrated with the third generation EST, and on the top right, you see this little kind of thing that goes around. That's got 60 of these cartridges that are in that hand on the top left. This again does filtration, preservation in situ, but it also can do some processing. It's gone off on 500 kilometer transects, and it goes to 300 meters depth, so you could actually get across the nutricline on those transects and dive and look around for the microbes. And then the Clio is out of here, Woods Hole from Max Sato and Chip Breyer, and it has to be deployed from a ship, but it's capable of going down very deep to thousands of meters depth. It takes larger volume samples as well. So I'm going to skip through this pretty quickly. Actually, I'm all right. This is Susanne Evans, who's been leading a lot of the deployments of the Roxy in recent years. And she's really done a phenomenal job of integrating it with the engineering team onto a lot of different vehicles. So here we've got an ROV on the left, we've got AutoSub 6000, which is the deeper AutoSub in the middle, and then the long-range AutoSub. This is the newer AutoSub 5 on the right. And it's also been in a tow body that's meant to be on an autonomous surface vehicle. So it's really kind of capable of being integrated with everything. The electronics and the communications are pretty straightforward. So it's been to 3,400 meters depth in 2021 in the North Atlantic, but then it also went to the Woodard Canyon and the Clarion-Clipperton Zone just this year, and that was its deepest dive at 4,700 meters. And I should say, a load of projects have funded this over the years, and because it's a lot of development. It's the first prototype, which is there with Robin on the bottom left, is a very different beast. And where have we gone with it? So these are just pictures of the ecosystem. So there's a coral area, and there's also an abyssal plain that's got polymetallic nodules. It's been in the surface as well. So in bold are the deep sea, and then it's done a lot of surface deployments on underway systems as well as on pontoons and moorings. And it filters 2 liters to 4 liters of water, and then you can recover that and do what you want with it. We're doing metabarcoding primarily, and quantitative PCR as well. It's been deployed for 24 hours, but it could be out for as long as you want. It's a biofouling issue, so it depends on your environment, really. Where we're going with it is, as part of Tech Oceans, we're going to do some demonstrations. So you develop these technologies, and then you do some validation in situ. But that validation actually is really more valuable if you have that sustained data set at that time series site, because now you know how to analyze your data and whether they make sense or if they're way off the mark. So this is in collaboration with Rafaela Casati and Daniele Iudicone and others at the NOAA Observatory. And that is an augmented observatory. They are already collecting metatranscriptomic data, metagenomics data, eDNA data, et cetera. So we can now put our auto sampler there. We're also deploying some genetic sensors there and kind of ground truth with their time series data. And then next year, we're going to go to the Canary Islands, and this has influence from the Saharan dust deposition, so it's off the coast of Africa. It also has a really nice bathymetry, and as part of Tech Oceans, we're using a load of vehicles, so we're going to be doing, it's not that autosub, but we'll use an autosub to get to depth, and we'll do some deep sea observations. This is combining chemistry sensors with the molecular detection and molecular sampling. It's going to be on an autonomous surface vehicle as well, and we're going to be deploying more chemical sensors on gliders. The objective here is just demonstration, but we're trying to get our scientific questions in line before this. So if you're interested in these regions and these areas, let me know. We're doing deep sea stuff with genetics and some benthic imaging, and then the surface ocean stuff. We're going to have this autonaut in the surface, but also one of the autonomous vehicles, so we'll be able to do the genetics in combination with the chemistry. The area is really interesting because there's a great eddy field just south of the Canary Islands. You get a lot of coastal influence of nitrate, and so that submerges, but you get a really dynamic field where you're going to see a nice gradient in chemistry and a nice gradient in the plankton. So where are things going? So I like Jamie. I don't know where Jamie Palter is if she's here, but I liked it. A couple days ago, she gave a fantastic flash talk, which was, I got to this quickly. This is an autonomous surface vehicle. We call them ASVs, and that's not whatever, a sequence variant. What does it stand for? So this is, I think, the one field where you might have issues saying that acronym, but yeah, so it is kind of the size. You could get them in different sizes. This is the blue bottle from OCEUS, and it uses wind, solar, and wave power to just propel itself. It's no fossil fuels. It can, because it's got all those options, it could actually traverse ocean basins. And it's got a huge area, so you could get these the size of a small boat, and you could put in loads of sensors. As long as they're happy to be in choppy water, that's the key. So you could get these to do geochemical measurements, physical measurements, and biological measurements at the same time over huge scales. And I think that's what we need to be doing, and how we need to kind of augment these sustained observatories. We end up going out to moorings quite a lot at NOAA, so we have our fixed point moorings. If we could send these out to do some higher resolution biological measurements in concert with those fundamental physical and chemical long time series measurements, we could add in the space component on top of the time series component. So that lab-on-chip chemical sensors are what ocean technology and engineering that's their expertise. This is a program that began with Matt Mollum, and he's a sensors person, and they're microfluidic. They're very small, but they actually measure a load of parameters relevant for oceanographers. As the drivers of the microbial activity, we need to kind of couple those measurements. On the bottom left, this is a transit that of 4 of these blue bottles, and they've been able to get across 400 nautical miles with no fossil fuels, no intervention. And I think if we combine that technology with the chemical measurements and the auto sampling, we could get at really large-scale measurements in short order. I just wanted to put a plug in as the drivers of the microbial activity. We have wet chemical sensors now in the Ocean Technology and Engineering group that measure all sorts of macronutrients, micronutrients, but also the carbonate parameters. A lot of these are now available commercially through Clearwater Sensors. There's a lot that's going to happen in the next few years. It's really an exciting time in the UK. So NERC are our funding agency, and they've committed to be net zero by 2040. Their biggest footprint is their ships, and so they want to use them smarter and maybe have less ship activity. But on top of that, we know that the autonomous systems actually have a lower carbon footprint. So they're really investing in what we could do differently with autonomy. So can we make better samplers, make different samplers, make better vehicles to combine with those samples. So they're really committed to, right now, to transforming how we do things with autonomy. And on a global scale, we really need to coordinate, and I know this is going to be a topic on in our panel, but coordinate measurements. So we have the Ocean Biodiversity Observing Network. This is an Ocean Decade program. These are all the sampling sites that are represented by the projects within the program. There's about 25 projects. This is kind of looking to make a global observing system, but what's required for that is more representation in areas that are not covered here, but also interoperability of our data sets together. We need to start standardizing across and see what kind of measurements we might do in collaboration that might be an additional to our time series data sets. This is to build into ecosystem models and deliver on policy. And I wanted to end by saying, I got in this because of the importance of microbes. Everybody who's a microbiologist understands their crucial role in biogeochemistry and in food webs. And it's really, it's interesting that something so tiny has such a huge impact. But I also was in Alvin, and we got to a hydrothermal vent area, and through the window of the Alvin, you could see so much diversity. There were little baby jellyfish and little, baby bivalves, all sorts of just plankton that, I mean, it was just mind-blowing. It was what really inspires you to, keep going. We know that this biodiversity decline is happening globally. It's mostly in terrestrial regions, but as we start pushing and pushing what we do in the ocean, we don't want to lose this gorgeous biodiversity, right? Imagine this picture here with, 80% less types of organisms. So I just wanted to leave it kind of on that note and thank the thanks to all my colleagues. I mean, I talked about a load of different work, but this is ocean technology and engineering on the top right, and this is the OBON group on the bottom left. So thank you.
Thank you, Julie. As we get set up for the lightning talks, if we have one quick question, maybe, from the audience.
I have a question about the Ascension sampler. You said you're enabling sort of citizen science to collect microbial water samples. How does, so if you have some person who isn't trained in microbial ecology handling techniques, how do you get the filters out of that or preserved in such a way that samples don't get corrupted before it ends up in the right -80 freezer?
Yeah, I think that's it. So the preservative is crucial, and yeah. So what has happened in areas where there isn't a -80 is that they've added on a preservative that they know how to interpret the data over time. So you can keep it at room temperature, but oh, I see what you're saying. No, you're right. So you need gloves and you need tweezers and a tube. And yeah, so you actually need to take them out. So they're 45 micron filters, as you would do on the deck of a ship in the lab.
Okay, thank you, Julie. We're going to move into a series of 5 lightning talks before the break. So the 5 speakers that we have are Camila Serra-Pompei, Aimee Neeley, Marco Corrales-Ugalde, Annette Govindarajan, and Carla Ferencia Berghoff. I will say that one of the big reasons to have lightning talks is to encourage people to go to posters that I think all of these people had. We're the last day. There isn't a poster session afterwards. However, there is a break afterwards. So at the break, if you hear something that you thought was really interesting, you want to talk to the lightning presenter more, please feel free to do that. You will see all of them right beforehand.
Hi, everyone. Today I'm going to be talking about backscattering and phytoplankton. And the reason why I got interested in backscattering was because normally with satellites, before at least, and now as well, we're using chlorophyll, but as a lot of you know, the chlorophyll to carbon ratio varies quite a lot. So it had been suggested that backscattering could be a good proxy to estimate directly phytoplankton carbon biomass. The problem is that the backscattering signal in the ocean also captures the signal of other components and not only phytoplankton. So we also have POC, minerals, heterotrophic bacteria, etc. Still, backscattering is used often in satellite algorithms, such as the CBPM model, primary production model, and it's often used to estimate, in general, phytoplankton carbon biomass. Here, I'm showing basically most of the data that exists in combination of phytoplankton carbon collected in the field with backscattering. And as you can see, there is not much data. And with these kind of algorithms that have been calibrated with this very few data, we pretend to estimate phytoplankton at the global scale. So I was wondering how good of a proxy can really be backscattering to estimate total phytoplankton carbon biomass in the ocean. And the way I did this was to first, I divided the project in 2 parts. The first one was a small review about the existing phytoplankton carbon algorithms. And in the other, I did a small modeling exercise. So since there is not much time, I will just center on the 2 of the main results. Here I'm showing, I compiled basically just most of the algorithms, which are basically simple linear regression. And I'm showing here backscattering in the x-axis and phytoplankton biomass in the y-axis. And as you can see, when you go below a given threshold of backscattering, these algorithms differ quite a lot between each other. And then I often get the question, yeah, but how often do you go below this threshold? And the answer is, it depends. It depends on the sensor. For example, I was playing with some Argo flow data, and actually in oligotrophic regions and winter in high latitudes, I could often go below this threshold. So there, depending on the algorithm that you choose, you will get very different phytoplankton carbon values. On the other hand, when I was using backscattering derived from the GIOP inversion algorithm using the MODIS sensor, I was almost never below this threshold. So if you use MODIS, backscattering derived from there, you might be fine with almost whichever of the algorithms that you choose. Next, for the modeling exercise, I was, okay, but what if we remove all these methodological issues? What if I had all the data in the world? How well would this linear regression be at estimating phytoplankton carbon in the entire world? And this, we can only do it with models. So I used the MIT GCM, which has also known often as a Darwin model, this biogeochemical component, and which has phytoplankton functional types and also all the inherent optical properties associated to each functional type. And so here I can explore the contribution of each component to backscattering. So I repeated exactly the same steps as the others, but here I have all the data, as I said. So again, backscattering, output from the model, and phytoplankton, again, also output from the model. And I said, okay, well, here I have all the real phytoplankton that comes out from the model. And here is the difference between the phytoplankton estimated using that linear regression and the model. On average, we get an error around 30% at the global scale. But as you can see, there are a lot of regional differences. So, well, here, that's where the fieldwork people come, and they should go and sample and test really whether these differences happen or not. As I said, this is just a modeling exercise. And again, this is just a lower threshold because we don't take into account all the methodological uncertainties. So just don't take it as like an absolute error that might happen by estimating backscattering. And with this, I'm done. Thank you very much.
Good morning. I'm going to present a non-researchy talk today. I'm going to be talking about the OPO working group, or the Operational Phytoplankton Observation Group, that I have to say very slowly, otherwise I reverse all the words. And this was recently funded by the OCB. So thank you, OCB, and those who chose to fund us. And this is co-led with myself and Nicole Poulton and Sophie Clayton. And we are very motivated and excited about this working group. Okay, so several other talks have already introduced imaging systems, so I don't think I have to go into that. But the point that I want to make here from this Lombard et al. 2019 paper, I love this figure, even though it's slightly outdated now because we have more instruments, shows the list of instruments that existed at the time that collect images of particles in phytoplankton. And then on the x-axis is the size ranges that those instruments collect of the particles. So we can see that we're hitting an era that we are collecting all these data from these instruments, and now is the time to develop a protocol of how we actually collect this data. And, you know, some of you might remember the Phytoplankton Taxonomic Working Group, where we developed standards and best practices for reporting these sorts of data. Now we're taking a step back. How are we all collecting these data? You know, a la the IOCCG protocols, NASA protocols, just to come to some sort of standardized way that we do these measurements. So thus, we created the OPO working group, and this QR code here is actually will take you directly to our flash page on the OCB website. And so our purpose is to develop standards and practices for the collection of particles and phytoplanktons from these imaging instruments. So let's think about the way we use these instruments. We collect data in different types of water, coastal versus oceanic. Someone on Monday mentioned sample volumes for omics. That's the same problem we have here. How much volume do we need to collect to truly characterize the population of where we're sampling? We deploy these instruments in various different ways. We have moorings, we have underway, we have profiling instruments. So we are doing this in all different ways that we kind of need to standardize how we're doing that. And then we also want to include the downstream processing. We're all doing this. We're collecting terabytes, probably gigabytes to terabytes of data. We're all doing this separately. Let's come together and make our lives a lot easier and come up with a way to do these things that we can all use. So our deliverables are going to be a protocol document of some sort. We're going to have a publication, hopefully get endorsement from the community, and we're also going to hold a town hall at OSM 2024, hopefully, if we're selected, if we submitted an abstract, so that we want the community involved. This is not built in a vacuum. We want everybody who does these measurements involved and provide their feedback. So that's super important to get everybody out there. We have 2 meetings scheduled. Well, we have virtual meetings, one coming up at the end of this month. We have an in-person meeting at Bigelow in August, and then we'll have a meeting at OSM 2024. So we are rocking and rolling, getting this working group going. And lastly, I just want to highlight the folks that are part of this working group. We had about 43, I think, applicants, amazing applicants. Unfortunately, we had to narrow that down to about 20 or so people. So these are the names and the faces of those who are selected to be part of the working group, some of which who are here today. Myself, of course, Nicole and Sophie, Emily, Marco, and Heidi are all here as well. So I'm super excited, we all are, to meet all these new faces and to come together with these motivated people to create these best standards of practices for these measurements. So I'll end with that. Thank you.
Thanks for the opportunity to present our new database on pelagic cycle structure from imaging systems. Oh, can I move around? Fine, I'll try not to move around then. Yeah, so the reason why we built this database is that we know since the 70s that there's an inverse relationship between the abundance of particles and the size of said particles. And when, well, these are log transformed, but the power law function that we can apply to these relationships usually has a slope or an exponent of around -1. However, there is intrinsic environmental and biological variability in marine systems, and that sometimes results in different pelagic communities of having like oscillations in that slope. So for instance, the community in blue has a less steep slope compared to the one in orange. So those values are known to be intrinsically related to all of those carbon and material flows that everybody's interested in here, right? So we can see, for instance, in this case, the carbon export at 120 meters is like increases when this slope like decreases or becomes less negative, just because there's more particles and things. Thanks, Camila, for that really cool study. So far, we have included data from the imaging flow cytobot, from the underwater vision profiler, and the SUSCAN. We have standardized, gridded, and averaged by time the data. The first release of this database is in Zenodo, and you can download the data by scanning that QR code. And an example of one of the products of this database is the normalized biovolume per size class, that in terms of biovolume. And right now, our products are instrument specific. When we do the global average to compare what we get for the different instruments, we see that there's like a nice, we're really surprised that it's a nice, like they're very close to each other, and these values of these exponents or the slopes are comparable to previous published studies and estimations of these slopes. And in general, this is the coverage of our data. You can see that most of the ICB data is restricted so far to the Northern Hemisphere. IFCB, SUSCAN, and UVP are more widely distributed. And the size of these circles represents the temporal coverage of the data. There's more temporal coverage in the Mediterranean and along the coast of the US. We are just starting to dig with the data, but right now we can do, say, a seasonal analysis of the magnitude of these exponents for different ocean basins. And we can see, for instance, that the North Atlantic and South Pacific tend to have less variability on these estimates compared to, for instance, the Arctic Ocean and the Mediterranean Sea. But we want more data. Everybody wants more data. So if at any point as a potential data user, you use this data and you want to see something else in this database, or you have data that you want to contribute to the database, we are really happy to talk about it. That's my email, and thank you so much.
Hi, everybody. My name is Annette Govindarajan. I'm at HUI. I'm going to talk about some environmental DNA sampling strategies for ocean observing. We have a lot of this work that I'm going to talk about described in a paper that recently came out in Oceanography magazine, if people are interested in more details, and acknowledging my co-authors on these works. Thanks. So these motivations for doing eDNA sampling and analysis for studying the ocean twilight zone or the mesopelagic is to look at what species, and specifically the focus of our work has been on animal species as opposed to microbes. But a lot of what we find could potentially be made applicable for microbes as well. So we're looking at processes like diel vertical migration, species depth distributions, and then what are the functional roles of those species. So when we think about eDNA sampling and analyses, we want to, it's a new approach, and applying it into these deeper waters is newer still. So we want to think about it like any other type of sensor data, really, where when we look at our results, how does that reflect the actual ecosystem? So if we look at the top part of this figure, right, we've got the various species in nature, in the actual environment. And then on the right-hand side, there's what the eDNA results may show us. Now, if we look in the middle, they may not necessarily align, right? So we want to think about how to interpret our data, what are the biases that go into this, right? And how can we improve on that and have a better understanding of our data? So I like to think about it in different categories. We have environmental factors like animal DNA shedding rates, how that eDNA moves around in the environment, how it's dispersed, and how quickly it decays. We have sampling strategies. How much volume do we sample? Where do we sample? How many replicates do we do? And then we have our laboratory factors, our various protocols and, excuse me, analysis approaches, our reference databases in which we assign a taxonomy. All of these things can influence our results and bias what we see from nature. So some highlights of the work we've done to try to understand this process. So one of the things we found is, especially for deeper water, as we go deeper, the eDNA in the environment decreases, right? So, so it's very important that we sample large volumes for animal DNA. The larger volumes that we find, the more that we sample, the more species that we find. We find that eDNA allows us to detect species that are missed by traditional sampling methods, okay? And so we can get a more comprehensive picture of the biodiversity. We do know that many species can avoid being caught by nets, so this isn't too surprising, but it does show the importance of using complementary eDNA analyses. One of the important factors with looking at eDNA is, does that signal represent the sampling location or somewhere upstream from that? And so we've also looked into that question. And so a modeling study that was done in my lab, led by Eiley Allen, shows that actually the DNA most likely remains in the vertical direction, pretty close to where it was shed. So that's good news if we want to infer vertical processes like diel vertical migration. We do see in our data sets that we do see vertical structure as a result of the eDNA. So this is our sampling technology, and we also heard about some of the different types of eDNA samplers out there. Our version that the technology that we've developed, led by Alan Adams, Dana Yerger, and myself, as well as others at HUI, takes large volume samples. It has a high flow rate, so we can get about 2 liters per minute, and that's important when we're sampling large volumes, and we don't want that integrated over a period of time. So the sampler also can go on a variety of different platforms, autonomous platforms, towed platforms, and we can apply that with adaptive sampling. So I'll end there, but that was really quick. If you have questions, reach out to me, or I'll see you in the break. Thank you.
So, hi, I'm Carla from Argentina, and I'm going to speak about marine ecological time series in the Uruguay Common Fisheries Zone. So we are a huge group, and I'm talking on behalf of all of these people. And so I'm gonna talk about a little bit about defining how highly productive and how strong sink area the Argentine Atlantic sector is. And its dynamic is really complex, mostly linked to biological signals. And this area, which is where the Argentinian Common Fisheries area is, it's really a biogeochemical transition region where both biological and thermal domains act at the same time. And this is where we are having our group is maintaining in situ observations of an ensemble of physical, biogeochemical, and biological in situ measurements of, for example, in biology of all plankton size community structure. And it's been almost 20 years, well, 23 years since we are running coastal time series stations called EPEA, Estación Permanente de Estudios Ambientales. We now have 161 cruises, so we cover really nice space variability, temporal variability. We also have the 3 coast to the shelf sections that we are still like adding new ones. We started from the coastal in Argentinian side, and we are added up now. And recently, we just started a citizen science diverse base coastal time series, which is called El Beril. The time is really short, so I'm gonna give a glimpse about EPEA and Coastal. So, okay. Kind of a heavy slide. So, coastal Argentinian coastal section started in the late 80s, and we have the knowledge of the surface seasonal cycle of chlorophyll, but nowadays, thanks to new additions of sensors as the PCO2 underway sensor fit in our ship, we now can see and understand the variability of biology in this area linked to phytoplankton size. And here you can see 2 different summer situations, a mid-summer situation and an early summer situation. And we can see that the green is chlorophyll, and the black is PCO2. And you can see how the variability is mostly linked to small size of phytoplankton. This is invaluable new information that we have in an area that is usually not being sampled. And another example that I'm going to show is our EPEA time series station, some preliminary results. We cover a lot of variables in this station, so I'm not going to show all of them, but some of them are being sampled, like chlorophyll, of course, and all phytoplankton sizes are being sampled since early 2000, but maybe not dissolved oxygen or carbon system measurements. They were incorporated in 2015. So what I did is to model all the variability. So we now know we understand the seasonal cycle and the temporal variability of most of the variables that we are being sampled, but not most of them. Some trends are really significant. Of course, we are experiencing an increase in chlorophyll and chlorophyll, small chlorophyll size linked to phytoplankton. So we know that is happening, but other trends that we model, for example, for the carbon system chemistry, it's not significant. So when I'm here today, is like eager to collaborate to help me on this. So, and also if you want to ask me some question about the citizen science project, we can talk on during the break. So thank you.
Thank you so much for those great lightning talks.
So to close out the session, we have 2 more talks and then a panel discussion. So our first talk of the last part of the session is Enrique Montes from AOML. That's the Miami lab, the NOAA lab in Miami. And so he's going to talk about from microbes to vertebrates, a marine biodiversity observation network for monitoring life in the sea. So take it away, Enrique.
Thank you very much, Luke, and thanks everybody to stay around. I'm super happy, and thanks to the organizers for inviting me to talk about the Marine Biodiversity Observation Network, which is a program that has been running for the past 7 or 8 years. And I'm super excited to talk about MBON at OCB. First of all, because I came to the first OCB in my first year of as a grad student, and I've been seeing the evolution of this program. And in many ways, because OCB and MBON share many elements of their philosophy of building communities of practice and building capacity as well, and bringing people together to solve really challenging scientific problems, right? So in this talk, sorry, I will, of course, start with a quick background so that you know where this is all coming from. I will provide a couple of case studies or examples of MBON implementations in the US and internationally. Some of the approaches that we're using, we already heard about eDNA imaging and so on. And I'm going to give you examples of how some of the MBON implementations are applying these techniques to understand how communities are changing over time, how we're integrating data with satellite remote sensing observations, and one slide on data management, and I think this is very important, but I promise I will not kill you by PowerPoint on this, and and examples of what we're doing at the international level. So, show of hands of how many of you know about the Ocean Biodiversity Information System. Okay, some of you. It's a great resource. It sits under UNESCO. It's by far the largest taxonomic data repository that exists out there. So if you're interested in ecology and species of interest for your science, this is a place to go. But the big problem is that if you plot all of the taxonomic records over the past 100 years on a map, looking at the upper 20 meters of the water column, you get something like this with major areas in the pelagic zone without a single observation. If you look at the coastal zone, you see that the data density increases, but it's highly variable from one continent to another. An important point here is that observations drop off drastically after about 210, and it's not because data is not being collected, it's simply because of delays of formatting the data and publishing the data in OBIS. So there is a big lag in data being incorporated into OBIS. And this is a paper from Emmett in which he shows that most of these observations are concentrated in the continental shelf and the slope, and there is the density in the deep ocean is really low. So the main motivation for MBON is to really tackle this challenge of increasing data coverage and the frequency of these observations, because in many instances, this is just a single snapshot of a sample that got collected somewhere and never repeated again. So under these circumstances, it's really hard to understand how biological communities are changing spatially and temporally. I like this map showing continuous plankton recorder surveys, which is possibly the longest ecological survey that exists out there. Data is not only scarce, but it's also geographically biased. These surveys have been concentrated in these areas, and there's almost nothing in some of the subtropical areas and tropics. So a big motivation for this program and for MBON as well is to really solve this issue. So with this in mind, I would say the overarching goal of MBON is to facilitate or create frameworks that increase data coverage, but also integrating these biological observations with physical data, biogeochemical data that is being collected by many frameworks at the international level under the Global Ocean Observing System, other networks, and to really concentrate on the interoperability and data access of taxonomic observations. So the way we're thinking about this is, of course, integrating historical and ongoing observations with new observations that use traditional techniques, NOAA techniques, that integrate with remote sensing, and thinking about ensuring that people have access to databases and data sets in a way that they can filter the data by taxonomy, by space and time, and be able to create maps that show distribution of interest, time series of species abundances, and also compute trends. And from these observations, then it's the natural progression is to develop indicators that then can inform large frameworks like the IPCC and others, right? To really see how the changing world is really happening, but at the taxonomic level. In the US, there are 6 programs currently ongoing that are actually part of this framework, 2 on the East Coast, in the Gulf of Maine, in the southeast of the US, 1 in the Gulf of Mexico, 3 on the West Coast, and 1 in the Chukchi Sea in the Arctic. But this is one example in this neck of the woods in which Jeff Runges is leading a team in which they're very interested in understanding changes in the distributions and foraging patterns of North Atlantic right whales, resulting from the rapid warming that the Gulf has been experiencing since about 2010. And what they're seeing is that the sightings of right whale abundances in the Bay of Fundy have been declining together with the concentrations of this, I would say, keystone species, the Calanus copepod, Calanus marchicus, which its abundance have been declining very steeply. And so now the whales are forced to leave the Gulf and go to feed up in the Gulf of St. Lawrence, where they are not as protected as in the Gulf of Maine. And so they are now at a higher risk of entanglement with fishing gear and being hit by ships. And this is a highly endangered species. So they're trying to use data collected by this program to understand how to better protect these animals. In the Central and Northern California, Francisco Chavez is leading a program that is basically integrating all of these programs. There's many programs on the West Coast, like cow coffee, all of the observations that are being collected by Embury, the ATN, or the Animal Telemetry Network. So they have an integration of programs that cover almost every single trophic level you can imagine, from microbes all the way up to the top predators, right? And they are developing very clever techniques to compute habitat compression indices that track the areal extent of what they call cool water habitat, which is important because it sustains important fisheries there, but also marine mammal populations. And they are saying that when this cool water habitat contracts, forage fish gets very close to the coast, the whales go after the forage fish, and then the whale mortality increases drastically in this area because they get entangled with fishing gear. Also in the Monterey Bay, we applied network analysis that were quite similar to the ones that Gwen talked about on Monday, in which we are using environmental DNA to understand when, or at least detect when species assemblages that are unique or they have certain affinities to environmental conditions emerge in this area, and when they shift to a different assemblage on a more or less seasonal basis. And in these analyses, this is a handful of a slide, but in this analysis, the shape of the symbol represents a taxon, and the size of that symbol represents the number of connections with other within that network, and it then allows you to understand when these, or at least identify based on the genomics that of the eDNA of the sample that you collect, when these sub-networks appear and disappear throughout a yearly cycle. But also, these type of analysis allow us to detect trophic interactions that were not identified previously, or possible trophic interactions, but also assess the overall biodiversity of a system. In the Pacific Northwest, Maria Kavanaugh is leading a team that is leveraging this Shimada surveys to sample the areas using a whole suite of technologies that we already heard about, the flow cytobots, the in situ ichthyoplankton imaging system, which captures the larger fraction, this big plankton in the area, of course, applying eDNA as well. But, for example, an interesting result is, unsurprisingly, is that the plume coming out of the Columbia River, they found that with IFTV, it's being dominated by, of course, large diatom, whereas more offshore waters here, they find the smaller picoeukaryotes dominating those phytoplankton communities. In my neck of the woods, in Florida, we've been conducting bi-monthly survey cruises that depart from Miami, go around the Florida Keys into Florida Bay and up on the West Florida shelf into the Gulf of Mexico. And we've been progressively occupying more stations towards the north because those areas have been affected by hypoxia and red tide blooms they're creating major shifts in the ecosystem here. And what we're doing here is we're integrating hydrography, trace metal, carbonate system, and net community production observations with nettos, imaging of the plankton, environmental DNA, but also traditional phytoplankton observations, absorption coefficients, biooptics as well, IOPs, and also trying to integrate with animal telemetry. There's our colleague, Neil Hammerschlag, that is looking at top predators distributions in the area. Now, of course, all of these observations are collected at a single point in time at a very specific location, and we do need to rely on a robust biogeographic framework to scale in situ observations to much broader spatial and temporal scales. And we've done this traditionally by borrowing concepts from landscape ecology, and from that, the long-course promises emerged, and we've been using them for a long time. It's a fantastic representation of ocean biomes, but the challenge here is that these are static. We don't know how they change over time. And so Maria Cavanaugh came up with a very clever, and others have done that as well, like Matt Oliver, very clever way of representing the biomes in the ocean using satellite remote sensing in a dynamic fashion. So she creates the dynamic pelagic seascapes by integrating ocean color and physical data using machine learning and a machine learning algorithm that renders these maps in which each of these colors represent a pelagic habitat or ecosystem or biome. And you can see that those change over time, they expand, they contract seasonally. The good thing also is that this product is operational, produced by NOAA Coast Watch. It yields 33 categories for different seascapes that include CIs. Each of these categories cover, or represent or show areas where they're very oligotrophic warm waters all the way up to nutrient-rich, very cool waters throughout the world. And the interesting or the value of doing this at the global scale is that if you have class number 12 on the South Atlantic and it's showing up somewhere in the North Pacific, these are water masses that have the same biochemical properties. In this study, we applied chemotaxonomic analysis of phytoplankton using HPLC, and we matched these observations with satellite seascapes, and we, of course, found that the classes that are representing oligotrophic waters are dominated by the smaller phytoplankton, picoplankton, Synechococcus, Fluorococcus, and of course these seascapes with colors close to yellow and red are seascapes that carry lots of nutrients, are very productive, and those seascapes tended to be dominated by the larger phytoplankton diatoms and so on. We're now migrating into the next trophic group, which are plankton, or I would say including also large phytoplankton using a continuous particle imaging and classification system, or CIPIX, that we try to get profiles on each of the CTD stations during these surveys. And unsurprisingly, we're also matching CIPIX measurements with satellite remote sensing. These are very preliminary results from the last two cruises where we matched the number of occurrences of these different groups that you see on the left with a seascape category, co-occurring seascape category, and we initially are starting to see that some groups tend to have certain type of affinity with a particular seascape class. For example, we see that trichodesmium is showing up in this class, which is tropical subtropical transition waters. It's a very low nutrient concentrations, very low biomass, whereas other groups seem to be more cosmopolitan. So we hope that with enough repetition, we can start characterizing the plankton assemblages in each of these seascape categories. This is work that is being led by Luke Thompson and his former postdoc, Sean Anderson, in which they went out on the GOMEC 4 expedition in 2021 to the Gulf of Mexico, where they collected eDNA measurements all over the Gulf. And now we matched those eDNA microbial identified communities with a co-occurring seascape category. And it seems like there's a good, that each of these sample seascapes seem to have very particular or unique microbial communities. And an interesting finding from these observations is that we see that seascape number 21, which corresponds to warm blooms and nutrients, which is the yellow seascape that you see around the Gulf of Mexico. We found very, very high relative abundances of Cariniaceae reeds, which are associated with Caronia brevis and these red tide bloom-forming dinoflagellates. And this is encouraging because if this is a truly robust association, then it means that we can use seascapes as an indicator for waters that may be at high risk of a red tide bloom at a particular time of the year. Please, I need to hurry up. This is my data management slide. And all of these observations are so cool. We do great science, but if we don't make sure that every taxonomic observation that we measure ends up at OVIS or one of these global repositories, then it's a miss, right? And so the MBON has been promoting hardly the adoption of the Darwin Core standard to format the data using these standardized vocabularies that use, for example, ecological metadata language for formatting your metadata. And we've provided R-based and Python tools that allow people to easily format data to international standards, make sure that your lat longs follow these agreed standards, that your species are identified with a taxonomic assignment from a accepted library like the World Register of Marine Biodiversity of Marine Species. And we've been training people to format the data in a way that then can be published either in OVIS or GBIF. And by taking these steps, with people should be able to, for example, render these interactive infographics by which you can click on an icon and it'll display a time series of, for example, the red snappers in the Florida Keys and see how they change over time. And it's a very clever way of synthesizing complex survey data into a simple report that a manager then can use to make a decision or inform their agencies of how these resources are changing spatially and temporally. And I'm going to close up with the efforts that we're doing at the international level. And I have to say that Marine Geo, and you already heard from Emmett on Monday about this, has been pioneering this effort for much longer. And we've been following their steps to really understand how we can implement these efforts with the global community. Marine Geo has been relying on the Tenenbaum observatories that they have in several parts of the world, but also partnering with people from all over by building capacity, by coordinating experiments that measure, for example, predation pressure, by applying standardized methods to estimate the percent cover of a seagrass bed. And they have been very successful at that. And we follow those steps in this other program in which we collaborate closely called the MBOM Pulse of All of the Americas. Yes. In which we have been also conducting trainings and developing standardized sampling protocols also in the coastal zone, because these are the areas where people have access to, right? Not many people around the world have nice ships like we have. And so we've been able to focus on the rocky intersoil, sandy beaches, and also training people on how to data science techniques so they can visualize and map their data, integrated with remote sensing, publishing the data in OBIS, of course. And once there, these larger frameworks like the Global Ocean Observing System and GeoBond can then use those observations to develop essential ocean variables and essential biodiversity variables. We've conducted a series of workshops in Brazil, Argentina, and Mexico, well attended, and more recently we did an activity that was super exciting with the park rangers of the marine national parks in Patagonia, in Argentina, Patagonia, where we trained them on how to collect benthic imagery using these simple photo quadrats, how to process the data using CoralNet, which is an artificial intelligence or a machine learning classifier that allows you to simply compute the percent cover of functional groups and render this type of indicator dashboards that, with enough repetition, can help them understand how their rocky shores are changing and why, because we know that many of these rocky shores are losing percent cover of very important functional groups like mussel beds very quickly, but they don't really know why. And I thank you for your attention, and I'll take your questions now or later.
Any questions?
Great talk. That was, yeah, it's where we need to go. It was exciting to see. The NOAA seascapes, I saw it said operationally run by NOAA. The seascapes kind of, is this a piece of software that people can use, or do we need to currently collaborate with folks at NOAA? And
no, it's even simpler than that. You can download the seascape data as a raster file. Basically, you can either download them as within 8-day integrations or monthly means, and it'll basically a flat file with the categories that are showing up in the area of interest, with a number assigned to it. Each number is a seascape class or a category. You can see the description of what these different categories mean in terms of their CDOM concentration, chlorophyll, temperature, and the physical variables as well. So you get an idea of the type of water masses that you're looking at, right? But it's very easy. There are tools we have developed our tools to have access to download the data directly on what's an IPA. There's MATLAB code. And all of this runs through ERDAP, NOAA ERDAP. And so there are lots of resources. So I encourage you to Google an application an R package that we developed with Ben Best and Maria called the Seascaper that will walk you through that process.
perfect, thanks.
Let's thank Enrique once more time. All right, next up is one of the fellow PIs from the BioGOSHIP project, which is Nicole Polton from Bigelow Laboratory for Ocean Sciences.
Thank you, Adam, and thank you to the organizers for inviting me. Well, and we've made it to the final talk of OCB 2023. Welcome, and I am excited to be here to tell you a little bit about myself and on the BioGOSHIP program. As Adam mentioned, I'm at Bigelow Laboratory and I direct the Center for Aquatic Cytometry. So I'll be representing a lot of scientists here. So I'm more of an expert on the flow cytometry and imaging side of things. But let's get started. We're bookending this workshop by talking about biodiversity at the beginning, and we're talking and completing that circuit here. Basically, what we know is that plankton or autotrophic phytoplankton are really the base of the oceanic and the food web, and they underpin really the blue economy, which is expected to be upwards or worth $3 trillion in the future. And so phytoplankton really mediate a lot of those biogeochemical cycles, as which we have discussed throughout this week, and we are looking at these very important elements. So biogeoship and other things are what we're really looking for here is looking at the metabolic potential and the activity of phytoplankton recognized as this is really an important knowledge gap. And we're trying to really understand how phytoplankton are impacted by climate change. And this baseline that we've been talking throughout the entire week is super important in trying to understand whether we want to look at CDR, for example, or whether we're looking at changes in climate. So we really have to have a baseline of this current state of the ecosystem. So we've seen this a couple times throughout this afternoon or this morning session. And what we have here is really the state of the global observation system, and this is May 2023. And although we have a lot of gliders, we have floats, we have a lot of these sustained shipboard observations, which are sort of in the orange in the background here. This is the repeat hydrography here. What we're seeing is that we're also missing quite a bit of the biology. Now that we have the BioArgo system here, we can actually start to really get at more of the biology in that component. We also saw this earlier. This is from OBIS, and looking at, and this is highlighted by Enrique and also by Paula, we also have a lot of records over the last 100 years, but we really have no idea what's going out on, or really have very limited knowledge of the open ocean and some of these fewer records that are taking place offshore. So where we're really interested in is trying to do more sustained and long-term and looking over maybe decades with regard to our ocean observations. So this is from the GOSHIP program, which are the Global Ocean Survey, Hydrographic Survey. And this is an international program, as mentioned earlier by Luke. And this really is a very important survey that's been going on for decades now. And these are, as I said, very sustained observations that are international, many of which are based from the US GOSHIP program. So what we're mostly interested in here is that this is primarily a program that has done chemical and hydrographic observations. And we are a team here that's really trying to add on the BioGOSHIP components, or the biological components. So whom we have here, we have Adam Martiny. These are the players and the team that we put together and led by Adam Martiny, but we are all working together here from all different aspects of the BioGOSHIP work. We have Harry Alexander, Sophie Clayton, Jason Graff, myself. And instrumental here has been OCB in trying to get this started and begun. So we also have Emmanuel Boss, and as I mentioned, Luke Thompson. And I'd be remiss if I did not say that the funding agencies really have brought this aspect, or this pilot program, which I'm going to be telling you about today, together through NOAA, NASA, and NSF. So as I mentioned, the vision of BioGOSHIP is really to develop this link between the physical chemical environment and the diversity of ocean plankton, and really understanding the biogeochemical roles in the context of a changing ocean. So what are some of these really key questions that we need to ask? And we've reiterated this throughout the entire week. And some of these things are, what are the global patterns of planktonic or taxonomy diversity or functional diversity? How do these physical processes affect plankton community composition and diversity? How do plankton modulate the global ocean biochemical cycles of carbon, nitrogen, and phosphorus? And what these key rates, how are we moving carbon throughout the system through ocean basins and biogeographic regions? And what are the global patterns in phytoplankton size spectrum? And how are we doing this? So you've heard from Julie, you've heard from Enrique. We have a lot of sensors and tools to do this in different approaches. And we are trying to combine this on board these ships using methods such as flow cytometry, image analysis, biooptics, and particulates and omics. So we are beginning this project with a pilot project, and we have chosen and added 2 lines that Adam Martini's group has done funded through the NSF. He was funded on sort of ships of opportunity to go out on some of these cruises. Those are designated in red. And the pilot projects that we've been going out on as of last year, the PO2 line in the Pacific, which is highlighted off over there and ends up over here. We just finished the A16N line, and now upcoming this summer is the IO5 line and then we have, or would like to, propose sections where we're looking at different aspects of on the black lines here. We have an Arctic section, another longitudinal section that's in the Pacific, and a few in the Southern Atlantic. And we're really trying to highlight areas that are really undersampled for this work. All right, so the BioGo ship, what are some of the core observations and how are we adding on? One of the key things that we need to do on these cruises is really we need to keep a low personnel footprint. So we are using a lot of these newer sensors, but not so much newer sensors, but also sensors that can be deployed through flow-through optics and as well as on the CTD. So what you have here is inline and CTD observations, and these are some of the tools that we're using. We have backscatter, chlorophyll CDOM. Some of these are relative to, as I say, with flow-through optics that would be very applicable to aligning with NASA or satellite validation. We also have fast rate repetition fluorometry. And as I just mentioned, I'm very much involved in the flow cytometry and the imaging flow cytometry. On the CTD, we've been hoping to add in things like the UVP, the underwater vision profiler, which is another sensor. And then back on land, this is where we're gathering all of the omics data and gathering, and as Julie mentioned earlier, we have a ton of samples and filtering that we do on board these cruises. So the acoustics is something that we really hope to add, which would really get at the metazoans in the system, and that would be with a shipboard EK80. So as I said, we need to keep our footprint pretty small on board the ship, and this is a picture sort of scanning sort of throughout the hydro lab on board the RB Ravel. And this is the BioGo ship underway sensors and sampling, where we have on the far side there areas where we're doing some filtration. We have the flow-through optics in the middle, as well as the flow cytometry set up here. And over here to the far right, we have a series of an area where we're doing a lot of the filtration, as I mentioned earlier. So what we're really interested in doing is we have to make sure that we're integrating between the BioGo ship and the Go ship tools and being able to use the hydrography as well as the deep elemental stocks and compare that and interact those together so that we are looking at the biodiversity, the biological stocks, as well as the sources and sinks. We're looking for new tracers that would potentially assist with some of the hydrographic observations that are here. And so that this would be able to link elemental cycles and also be able to really validate remote sensing protocols or products. So I'm going to go over the next few slides. I'm going to be talking through quite a bit of the initial or preliminary data from our first occupation, which took place in 2022. So this is some data actually from the actual Go ship Go ship line. And what we have here are temperature, nitrate, and phosphate. And you can see that distribution across the entire basin in the Pacific. And you can see it from down below here is a chart which basically where the cruise line goes from Japan over to the upwelling area off the coast of California here. And what you can see here is the nitric line, which is the 1 micromolar line, and you can actually see how the depth and how that changes over the course of the transect, as well as the phosphate that you see here. And I'm going to be, if you can remember or look at this, and this is going to be relative to some of the biology that we're looking at here. So you can then, through the flow-through optics, or excuse me, with the flow-through system that we have on board, we're continuously, not always, but every 3 times a day, we are actually collecting POC and PON throughout the day and on filters and analyzing that. And we can then look at the stoichiometry. And some of this work was also for other cruises is actually presented by Adam Fagan, a student of Adam Martiny earlier as a poster. And what we can really look at is the variation, and you can see little variation. We are in the subtropical gyre, but we have high resolution data here, and we can actually see the C to N ratio change subtly in areas, or is actually above the Redfield ratio of 6, so that you can actually see that there's areas of potential nitrogen limitation. So something else, one of the main questions are some of the patterns of phytoplankton taxonomic diversity. And we're looking at here is HPLC data, where we are looking at the total chlorophyll A. And this, I must say, was all processed via NASA. And I just wanted to highlight that in terms of our CTD profiles, we've been given the opportunity to have an additional cast in addition to the ghost ship casts. And you can sort of see that distributed across this entire transect here, where we see at approximately 40 meters, as well as at about 100 meters, we are collecting HPLC measurements as well as omics at those depths. And then with the bio-Argo floats that are being deployed on these actual transects by the ghost ship team, you can see that there's a more higher resolution data that's being collected during those float casts. So when those are, so we're actually validating the BC Argo floats as those are being deployed on board. So you can see across the surface, this is continuous data that's being collected at different times of the day. And these HPLC samples have been monitored, and you can see a little bit of the Kuroshio, where you see an elevated chlorophyll on the far western side of the basin. And on the eastern side, you can see the upwelling zone that's predominantly near the California coast. So let's take a little bit of a deeper dive into the HPLC data now. And this is an analysis that's literally hot off the press. We got it this week. So we're really, this is in real time processing some of this data. And we're looking, was developed by Sasha Kramer. And this is an empirical orthogonal functional analysis, which was developed by Sasha Kramer and Dave Siegel. And they are looking at a statistical variability of the data, and they're analyzing it using this empirical orthogonal functional analysis. And what we're doing is looking at ratios between certain pigments to total chlorophyll and how that's varying. And we're looking at different modes within the data variability, and those modes associate with different phytoplankton types. So in the upper graph here, you can see on the far side, everything else plus divinyl chlorophyll B. Now, Prochlorococcus is known to only contain divinyl chlorophyll. It's really the only organism we know that has divinyl chlorophyll. There's A and B. And it's the ratio of those two, or the differences between the variability within those pigments, you can actually see the distribution potentially of Prochlorococcus. This is across the top here. And you can see the blue is indicating a decrease in that magnitude of that mode, indicating the presence of Prochlorococcus. So as you go across the basin here, you can see that there's a deepening in the presence of Prochlorococcus at depth. We also saw that the nitric line deepened there as well. And then you can also see in this plot here, the sort of a different pigment analysis, where we're looking at more of the ratios that identify or a mode where we're looking at the diatoms and dinoflagellates within the sample. And so the presence or absent, where you see a positive mode magnitude, you're going to see more of those organisms present. And we can see here in the on the upwelling zone over here, where there's more of a presence. And you can see a few within the surface, but very limited. but you can see that it's primarily a Prochlorococcus or a cyanobacterial dominated community here in the subtropical gyre. So using this HPLC data and other data, especially the fluorescence that we're collecting through our optics measurements, we can really see how this potentially could relate to validation protocols for NASA and PACE with their new mission up and coming. And what we have here is a relative data density. It's the blue you see in the background here of what is actually in the CBAS data set or online. And so what you can actually see here is that there's areas of limited coverage, and in areas of the Southern Ocean, we also have the Indian Ocean and the Pacific. And the lines that you see on this map are where the BioGoShip program will most likely collect data for validation, ideally. Paula very kindly introduced this slide already, pretty much. But what we're looking at here is what is the metabolic potential and the global patterns of nutrient limitation. And this is specifically looking at nutrient stress using metagenomics and Prochlorococcus. And this is work by Ustick and Larkin, and Larkin is one of our session organizers here, as well as from Adam Martini's group. This was a science paper that was really looking at how you could use these metagenomic data to really analyze and interpret nutrient stress amongst Prochlorococcus and maybe identify regions of nutrient stress, different regions of nutrient stress. So where P limitation is taking place versus nitrogen limitation, and where there's areas of co-limitation taking place. So what we have here are cruises. These are both BioGoShip cruises. These actually have some geotraces or biogeotraces cruises as well. And you can see the coverage here, but you see areas where Prochlorococcus is nitrogen or co-limited, and areas where they're iron limited or iron and phosphorus co-limitation together. So continuing on with the phytoplankton diversity and to our story earlier in the week where we had and we discussed quite a bit about biodiversity, and you can actually see here the data that's been collected from these about 1,000 different surface samples. And here's the distribution similar to our previous plot here, taking this similar data, but looking at the entire bacterial ocean diversity here. We've actually assessed that using the metagenomic data and looking at the Shannon's index. You can see a scale here, and you can see the different cruise tracks, and we've mapped that. And really what with the overall conclusions that we've found here is that nutrient stress and the frequency of Prochlorococcus is a very good indicator or a predictor of bacterial diversity in the system. And you can see that here where Prochlorococcus and Shannon's division work index have a high correlation with one another. Almost done. So in addition to that, speaking of Prochlorococcus and Synechococcus, with the high resolution flow cytometry data, similar to data that Heidi Sossik's group and the LTERs that are here off the northeast of New England or at MVCO, what we're hoping to do is really get at rates. And that's another thing that in terms of biological measurements that we are really lacking right now. So what we're trying to do here is look at the daily cell size in terms of, since we're doing continuous sampling here, this is actually every 10 minutes. We're actually binning this by hour here, and this is the estimated spherical diameter. And you can actually see that there's a trend, or excuse me, an up and down cycle, and that's representing the division rate. So the goal here, this is very preliminary, was to derive rates of division and carbon uptake from the system here based on these cyanobacterial populations. And again, it's another add-on to the diversity that we'll be able to measure, as well as the rates that they will get at their growth. And finally, we have really what are the patterns, and this is something, patterns of plankton size distribution. This is brought up by Marcos and Enrique, and really looking at the slopes of these particle size distributions over time. So this is literally off the press because we just only have analyzed the very beginning of the P2 analysis, and this is a credit to Stephanie O'Daly up at UAF in Alaska, and working with, or had been working with Andrew McDonald up there. And on board this cruise, we had both an underwater vision profiler and an imaging flow cytobot. And what we have here is at the surface, you can see that there's a broad, just even going from the Kuroshio out into the open ocean, within the size class of a half micron, and this is particles per liter. What we're actually seeing here is quite a size distribution or range in size distribution and particle load. So with that, I just want to mention that we have, we're doing a lot of, in addition to data collection and adding to the pool, we're also doing data standardization and integration. So what we have here is we have a lot of technologies that are constantly changing, such as extraction methods for molecular work, as well as new imaging techniques that need to be standardized or intercalibrated. So there's a lot of things like that. So there's no really agreed upon standard, and that's why we have things like the OPO working group. We're trying to come up with methodologies. And then as we have here with data integration, we're always trying, how are we going to bring all this data together? And that's a big challenge. And a lot of that's been sorted out by the GoShip group, but we're also working on how we're going to integrate molecular data with imaging data and bringing that together, and that's going to be a huge challenge as well. So in terms of the standardization, in terms of DNA sequencing and extraction, what we did here is we actually did an intercalibration of the metagenomic extractions, and this is a sour mash fingerprint plot right here, where we were testing 5 different methods, and what we have here is different laboratories, different laboratory groups, such as Adam Martini's group at UC Irvine. We also have NOAA AOML standard as well, and the actual extraction method that was used on the PO2. And what we basically, the summary of this plot is that really there does not seem to be any bias towards one method or the other. You can actually see across this plot, every row that's going across there, there's 2 samples, Miami and Newport Beach. They're color-coded. I'm going to go over here. And so this is the Miami group, and this is the Newport group, and each row represents a method that was tested, and these are the color-coded methods right here. And what you can see here is that the similarity is between all of the methods is very similar. So what we're finding is that we are doing these intercomparisons and cross-cruise comparisons. So hopefully that if someone's using a different method on one cruise, they can be intercomparable. So in summary, sustained biological parameters are chronically undersampled, and that's sort of been the highlight of this entire week. And it's really specifically for space and time, but also for a lot of these essential ocean variables. And we have a lot of mature technologies that can be used and to really make these observations, but we need to be consistent and we need to be routine about that. But we don't want to limit that. We want to also be testing and constantly keep decreasing our person footprint on some of these ships by incorporating a lot of new biological sensors, testing those sensors, and applying different sampling technologies. And as I just mentioned with data integration, we would also need to do a lot of QA and QC to really understand the intercalibration of these different protocols that we're using, and to make sure that accessing the data is fair, consistent with data policies, and again, with that whole entire access in terms of driving this new model interpretation and in increasing data science, but also doing a lot more of this Cal and Val in terms of QC. So what we're trying to do here is really what we need are these sustained biological observations. It's been something that's been lacking, and I think with the GoShip program, we can truly add on a critical element by including the biological component to something that's been done for multiple decades. So with that, I would take any questions, or we can go straight to panel. Thank you so much.
Thanks so much, Nicole. I think we're going to go straight to the panel, if that's okay. So I'm I'm going to get us started with some of these questions, and then we can open it up and have an open discussion about all of this. So I'm going to combine some of these into sort of categories in the interest of time. But the first question is, why do we need sustained observations of ocean biology? You've all kind of gone over that, but specifically, you know, why do we need them? What are the barriers? And also, is there an alternate viewpoint to committing to sustained observations? So I'm asking you to talk a little bit both about the positives of why we need it and a little bit about the negatives of what are the barriers or what are some alternatives. Okay, with that, I'll open it up. Does anyone want to get started? Paul.
Thank you for the question. So a couple of things. You know, I feel like, why are we in biology? Because if we wanted easy, we would have gone into physics. Sorry, kidding, sort of. When I started at NASA headquarters, there was one other woman there, and she was the other biologist. We call her the other woman, my mentor, Diane Wickland, and she always said that to me. She's like, if we wanted easy, we would have done something else. Couple things. First of all, time series in general are very challenging to sell, but I would say two things to the doubters or questioners. Number one is it allows us to explore in the temporal dimension, right? That is a dimension that is important over time. It allows us to track trends also, and we've talked about that quite a bit today, so that's very important. I think the other piece I just want to acknowledge is Nicole, among others here, gave a beautiful talk on answering the question I completely botched that Adam Martiny asked me this morning, which was about what observations do we need? And it occurred to me, specifically for Nicole's talk, that as an operator of the academic fleet and having worked very closely over my career with philanthropy and other nations, what BioGoShip is thinking about as standard observations really should be advocated for as standard on any cruises going out, right? So that will improve our capability to have that exploration, to look at those long-term trends, and to validate the multi-billion-dollar observations that are being made in Earth-observing satellites.
Agreed. No, I actually, what you just mentioned about having this actually be on all vessels, I think is because, as I mentioned, one of the goals of BioGoShip was to try to keep. We had in these last two cruises only two people working, one person working a 12-hour shift and the other working another 12-hour shift. But you could think of the flow-through optics, these new sensors, although they also need validation and intercalibration. But you could also see those being placed on ships, and if they all had flow-through systems, and most of them do, it's just adding on some of these additional sensors. I mean, I think the biggest, in terms of omics, it's a lot of filtering. But as Julie mentioned, there's probably tools where we can maybe automate this a bit more, where it wouldn't have to have a person physically sitting there filtering.
The other thing is that I think when we talked about all our new tools, we've been working with gene sensors as well, and we've been working with autosamplers and getting meta-transcriptomics data from there. What was key to that, I'd have some really nice data and think, oh, it looks like phosphate's driving X. And is that something that I should expect from this area? And it was something that had been seen again and again in the historic data. So you can do these short-term process studies. You could do things with automated sensors, but these one-off cruises, you end up making a lot more of your data when you could scale up with those historic data sets.
I always like a comment from my former advisor, Frank Miller Carter, saying that nobody questions the fact that, meteorological data, temperatures, precipitation, etc., is being collected all over the world and being shared on the spot right away. And from that, we all benefit from making sure that yesterday night we would not get like hammered by the rain, right? So it's a given, right? We need to think about biological observations in the same way, and that requires shifting the mindset to the idea of making sure that the biological data that we're all collecting become publicly available as soon as possible. And the challenge, more than what you were saying, Paula, that biology is freaking difficult, is convincing the agencies to adopt these new technologies, traditional methods to operationalize the observations of life in the sea. We in academia like to use the term time series to talk about the M word, or the monitoring, which we don't like to use, but truly, if we want to understand how the plankton, all of these ecosystem services that we extract from the ocean are changing spatially and temporally, and we can make predictions, we do need to convince agencies to implement monitoring across trophic levels all over the world and make a big investment in this.
We have one in our audience. Heather McNair, University of Rhode Island. And Nicole, those transects that you presented are amazing. For like the underway data is amazing, and then the points of POC, PON, HPLC, it seems like one of the challenges is that downstream. Like we can say we need to add these to every cruise that goes out, but Sasha says there's one person and one instrument at NASA that does all those HPLC things. So it seems like a much bigger infrastructure issue too.
I would say definitely. So, I mean, we would have to maybe branch out from having one place for all of these HPLC samples are run, especially if we're going to be collecting them at such high resolution. And we may not need them at that high resolution too, we may find, so, but if we're trying to be validating, a very expensive satellite that's going overhead, you're going to want that high resolution, especially, one of those time points is always at the noon 1 hour time frame. So it would always be, hitting that damn time point every day. So, and that would be the idea of the flow-through system in that sense. But yeah, I agree. There's many labs, there's many different techniques. Even I was thinking more of the POC, POM in terms of volume filtered, things like that. That's different amongst different organisms, different laboratories. So if we could find maybe central facilities where these could be run. I mean, I run a center myself for flow cytometry because those are really limiting. I mean, there are instrumentations that are coming more online now, but I think we could do similar. I mean, we have like nutrient analysis facilities, but maybe they could all be intercalibrated. But yes, it would require a lot more infrastructure.
Another up front. It's Susanne Craig, NASA Goddard. So looking at the suite of instruments and observations that are made on, if I use the BioGo ships as an example, they're amazing. They really give you a holistic view of what's happening at so many different stages in the whole ecosystem. As someone who works for a space agency that's about to launch a ocean color satellite, the one glaring omission to that beautiful suite of measurements was in situ measurements of ocean color or reflectance. That's our standard radiometric product that we measure from space, and then everything else is derived from it. And it's actually not a super easy measurement to make, but there are solutions like automated kind of roboticized positioning of the sensors that it could make it person-free. So from my little myopic corner of the world, I would love to see more global coverage of in situ ocean color. Yeah, we do ocean color from space, but it's also challenging to get. And really the gold standard is high quality in situ measurements of ocean color. And that would just be an absolutely amazing data set to have.
you know, I don't think we're limiting. We would love to add more to, so, and we have actually proposed to do more, but this is where we're at right now. But I agree. I mean, those are sorts of, we would love to do a lot more sensor development on board, but also working with these ships of opportunity that are working 24/7, and we've been graciously given additional cast to collect biological samples for the volume that's needed to do for metagenomic analysis.
Just to sell it a little bit more, those types of ocean color systems that I'm talking about, they don't need any casts, and
Right.
they can just be stuck on a boat.
On a boat, right?
Yeah,
no.
yeah.
Right. So it's a limit of instrumentation right now.
Absolutely.
So it's not
Yes.
easily added.
Thank you.
Okay.
One question in the audience, and then we have some online.
Okay.
Go ahead.
Chitek Lee from Korea. I'd like to combine this sustained observation of biology to carbon cycle. I spoke with a couple of global scale carbon modelers. They do not accurately represent the biological processes. So the pressing question, if I feel that biologists are very well equipped in terms of collecting really good data. If the pressing question is that how biology influenced the oceanic uptake of the CO2 in a changing world, a changing climate. So, for instance, like how MPP has changed over time, and also where, and also what fraction of the MPP really truly exported in the interior? Where are the hotspots? What are the driving mechanisms for remineralization of organic matter, which depth range, or dissolution of calcium carbonate? All those factors, I think biologists can provide really useful data, and then those data can naturally feed it into the global scale model. By doing that, I think the modelers, in conjunction with the biologists, can address how biology is to truly influence the future oceanic uptake of the CO2. I think some of the research should focus on these pressing questions. It's my personal opinion.
Any thoughts, panelists or our lightning speakers?
Thanks. Can I add something to you? I was thinking of the research questions each time series has, which it might be different because of the history of the time series. You were talking about like you need more biology measurements. Our time series had more biology measurements before having biogeochemical like carbon system measurements. So it depends on the history and the question that it's below that. So I think we need to harmonize that and we think about what we need in the future. Like, I mean, you cannot throw out the story of the time series. Maybe we focus on different things. So I don't think we can need to harmonize, maybe in open water, but in coastal waters, it's more dynamical. So that's something that I want to say.
And so on in response to the carbon question, I mean, I think people are I know people are doing it. It's just what our session was around is about in situ observations and ocean biology in general. But maybe we're not the right. There's people that use size classes and do things to better inform the models. So better capture that those transformations. But we also, when we're capturing the metatranscriptomics data, we are looking for the kind of the drivers of the change. So in response to a gradient that might be a gradient representative of future ocean change, you can look in the current ecosystems and kind of make interpretations that way. That's how the environmental microbiologists tend to address it. But.
Yeah, we have a project I'm working with Emily Osborne on a Gulf of Mexico where we have sediment traps, and we can measure the biological community at the surface as well as in these sediment traps that's collected over, now it's going to be multiple years, every 2 weeks or so. So we can see, how things are exported from the surface down to the deep mesopelagic. And so that kind of sustained observing, I think, is addressing some of the carbon export questions, at least.
I also, I think want to highlight the talks, and I really appreciated the talks given by Camila and Marco today about how we can better quantify things like plankton biomass and plankton size structure, and take those in situ quantifications and then use them to validate things like the satellite-based, right, plankton size class estimates, right? And so if we can improve the global coverage of those types of measurements, then that also better improves those satellite models, and in turn can improve the Earth system models, right? So I think we did hear from some people today who are doing some of that active work. I think it comes down to identifying, the regions where we most need those measurements to happen. We have a couple of questions online.
Yes, so the first question, I believe, was for Nicole, asking if red tide, oh wait, no, I'm not sure. Was it for Enrique? Does red tide correlate positively or negatively with benthic vegetation cover?
Wow, I don't know. Well, I don't know if you remember in one of my slides where I'm showing the map of Florida with stations along the Florida Keys and changing in color towards the north on the Gulf side. That we've been extending those cruises northward because because of these massive red tide blooms from Carenia brevis that we've been observing over the past few years. The additional complication of those red tide blooms is that they induce hypoxic conditions that may affect or likely affect very important seagrass beds in the area and all the macrofauna that live on them. So more than thinking of the benthic influence on the red tides, which there's lots of hypotheses on how this is maybe happening at the cyst levels and resting stages of these dinoflagellates that may be dormant in these habitats for a long time, and then they wake up and form blooms. These are things that are being actively studied at the moment, but I don't think there is solid conclusion along those lines. But anyhow, it's a very important point, and this is something that, at least for that area and many other parts of the world, is something that needs to be resolved sooner rather than later because of the major impacts these blooms have.
Another question. Benthic observations always seem to lag pelagic by a lot. Given that many vulnerable species are in the benthos, what are the panel's thoughts about new technologies that can help automate benthic observations of the global ocean?
Can try to get very deep, if that's what you're thinking. Yeah, so that's actually really why we did that depth. So let me start at the start. So a lot of people, actually a couple of people in this audience were on a cruise with me when we had a underway sampling of our automated eDNA sampler. It was fine, and it actually worked as intended. And when we came back, the first people who were interested were benthic ecologists. And so yeah, so we put on a pressure housing, redeveloped it, made a next generation that's good for deeper areas. But that all of those demonstrations were eDNA oriented, but also looking at the benthic microbiology, but in concert with the ROV imaging, in concert with the AUV acoustics, and even like vertically integrated acoustic data of coral communities. And so the objective is really to start. I mean, we've got areas that we monitor on a 6-month basis for marine protected areas with Vrilla Huvene at NOAA. And the idea is to go in there year after year and kind of get those sustained observations. It's hard to deploy and leave it there because it's hard to get back, right? You need to get back out there and get it. So landers are also an option, but they have to have that longevity, and the sampler has to be rated for depth. But I know Annette's got thoughts on this if you want to say something.
We have some work on putting a sampler on lander in progress, so hopefully I can report more on that soon, but we're thinking along those lines too.
Yeah, I actually have a bit of a generic question or something, but I'm interested in rates, and we tend to estimate much more stocks than rates. So I'm thinking, how can we try to derive rates from those stock measurements, and how far are we on that? So for example, using omics, perhaps the concentration of certain proteins or enzymes, for example, how can these be good proxies for rates, or how can we use, so you showed the flow through, the flow cytometry, but that only gives you about the picoplankton. How can we extend that to the larger guys, for example, maybe with backscattering? So how much investment is being done on deriving those rates from stocks that we can incorporate in the, for example, the Go sheep or other measurements? I don't know if anyone.
Sometimes it's a good question.
I think we measure what we can measure with the methods we have, and it's a very tricky thing. I mean, without doing, bottle incubations where you can label or pause certain processes or things like this. Its potential is something we could add to, to add an experimental component to a Go ship kind of program, but I think it's kind of tricky, yeah. But I think the flow cytometry example, the best thing we have.
Right now, but I mean, I guess in the open ocean where those will be dominating communities, so it would be, but still, I mean, we're lacking information about the upper trophic levels.
There's also some, at least for, so this doesn't help with the upper trophic levels, but there's also some, metagenomic data-based analyses that we can do, like metagenomically estimated replication rates. Similarly, that's only for bacteria, but I know there's some labs out there that are sort of developing ways that you might do that using mitochondrial DNA and chlorophyll components. And so there's also been some creative thought and work on how to use omics to estimate replication rates, which is exciting, but then also is more powerful if you can validate it with things like these flow cytometry-based growth metrics. And so it's an area where if we can, I think it's an active area of research of where we can find good crossovers between the omics and the flow cytometry, and paired with some of the more traditional primary production and uptake type measurements. I personally would be interested in seeing where that goes in the future. Adam.
I just want also to say that I saw a lot of really nice posters this week with people developing a whole variety of sort of underway techniques for measuring net community production, nitrogen fixation, and then like from isotopes and ratios of different elements in the surface ocean. I think there's a lot of interest in like measuring rates while avoiding to have to bring it in a bottle and incubate. It seems like there will be a lot of new technologies in the next couple of years.
Yeah, thinking on to that, I mean, it's not something that we're doing on like BioGoShip, but we just developed a method with flow cytometry to look at individual cell respiration rates. So using a redox sensor green and calibrating that to known respiration rates. So there are, I think, tools and methods being created that could be used. And we, summed up all the single cells, but then that's, again, really only looking at one population again, right? So, but in these bacterial communities, you could look at bacterial respiration rates and then add those rather than having to do these, 24-hour incubations. Or there was a great poster, I think, by Melissa Cohn, and she was presenting work with optodes that are on the export cruises. So I see those being added. So I'm trying to get at respiration.
So we've got about 15 minutes left, and I want to make sure I think we've touched on a lot of the questions that are up there, but I want to follow Enrique and take a little time for data. So I want to make sure we get to this question first and then open it back up to audience questions. But I wanted to ask, what is your biggest challenge, but also your biggest opportunity with regards to biological data management and data sharing? And when thinking about the opportunities, you might think about what new technologies, working groups, venue systems, et cetera, can help scientists achieve maximal data sharing and fair principles at minimal complexity, cost, and time investment.
Okay, buckle up. This is 45 minutes. Thanks for the question, really, because a very core element of the Marine Biodiversity Observation Network has been around the idea of managing data in a sustainable way and raising the awareness, the importance of doing this to really tackle some of the very difficult questions that you were asking before. How we combine biology with everything else that we collect, physical, biogeochemical, because this is the only way we're going to see what the role microbes, secondary producers, upper trophic levels have on carbon cycling and climate. If we don't make sure that biological taxonomic data becomes interoperable with these other databases and data sets, there's no way we're going to get there. So the big challenge to doing that is that you're a PhD student, you're gathering your data, super valuable, and you don't want to share it right away because your dissertation depends on that. Once you graduate and you publish your data, you should try to see how quickly you can push it to these open access repositories so that others can use it. Your papers will be read and you'll have a lot of fun talking about them, but papers disappear over time. But your data, that's the true contribution that each of us are going to do to our community. The challenge is that this requires skills, data science skills that we need to gather, and that takes time. It's a steep learning curve, but the opportunities are that there are already lots of tools out there that can make our lives very easy. If you use Python, then you can rely on pandas, packages that can help you reformat your data, your timestamp, your that longs very easily. You can allow you to merge columns here and there, filter your in many ways. The same with R. And learning to use those tools, I would say it's relatively easy, especially for people that are more computer savvy. And I think that we're moving in that direction. Lost my train of thought, but the tools are out there. If you look at the Ocean Biodiversity Information System, they have a whole toolbox of R code and Python-based script that can help you manage your data effectively, align it to the Darwin Core standard, and getting it published. An additional benefit to doing that is that every time you publish, your data set gets a DOI. So you probably, hopefully, will get cited for that data set, and that is good for you if you're pursuing a tenure track or academic career. And I think this needs to, I am advocating for this as a community engagement. We need to volunteer to doing this as an obligation, right? As something that needs to be done, because otherwise it's a missed opportunity. And I hope I answered to some extent your question.
want to quickly add to that. I'll just use this. That I think there are a lot of opportunities for actually increasing the training because with things like autonomous sensors, and you're going to have a data deluge, and you end up on top of that having people who really want the data, like with digital twins, and so that training piece between the gathering and the uploading of the data or formatting and all of this, I think that that's just absolutely something that we need to emphasize and yeah, just push forward. But it's going to happen, I'm sure. I haven't talked to people about it, but I'm sure I'm not the only one thinking about this.
Yeah, I mean, we're doing this with the omics, like we're developing data management guides, templates, also some software tools to be able to convert from between different formats, like to the Genomic Standards Consortium, MixS or Darwin Core. And, you know, I think the databases are improving, and, you know, someone mentioned ASVs, the other kind of ASVs, but there's also the sequence ASVs, which are the exact sequence that represents the taxon, which is a great way to preserve that fine scale resolution of the eDNA or the microbiome data. So lots of things, but I think the challenges are to truly integrate and get things in the same database. We still don't have databases that really work for us for the omics data. We have GenBank, which is a repository, but it's not you can't go and like do an analysis there or ask a question. So that's where we need to go. And I think between MBON and BioGoship and some of these programs that I have the luxury to be involved with, like we're gonna really try to get that done in the next few years. And luckily, you know, we have support from NOAA and we have support from the community to do that kind of stuff. So I'm pretty optimistic.
Okay.
So I feel like Enrique and all the folks here maybe stared into my soul about the whole angst-ridden process of data collation. I'm funded right now to do exactly that, try and put reflectance, which I'm interested in, ocean color, put that in a commonly standardized format database along with IFCB data, maybe some genetic or molecular data when we have it. And it's been a bear. It's been a bear, and I hired a data scientist to do it because I know it's a data science problem. And I've had these exact conversations with, you know, several different people. There are some barriers for folks getting their data into these standardized databases, and I think you've already said we can embrace the data science world and work with them to write as like APIs that are going to take the data, standardize it, and put it in as painless a fashion as possible. But I have this conversation across every discipline. I sat with Woody the other week at the NASA meeting, and he's like, we're trying to do the same thing, and it's this huge task. So I think if anything comes out of today's conversations, maybe we can start to collectively develop some guidelines and ways forward to do this across all of our different disciplines. Because I'm now 2 years into a project, and my data scientist is still working on these types of problems, and we haven't gotten our teeth into the actual science yet. And all of these wonderful technologies that you all described today, an absolute deluge of feature-rich data that we just need to get on with the task of exploiting. So, yeah.
Can I add one thing to that real quick? Sorry.
Yeah.
I'm not sure which of you said it, and I totally agree, but there was a comment made about the NASA HPLC data and one laboratory running that and submitting those data, you know, on a timeline to the CBAS database. That I did not make that decision lightly 20 years ago when we chose to do that. The challenge, it's a phenomenal training opportunity, you know, to have distributed labs doing that kind of work, no matter what the data, whatever the instrument, the laboratory process is. That's really important for training our future workforce. However, there was tremendous resistance to being held to certain standards and protocols at the time. Okay, so garbage in, garbage out, you guys. So we're going to have to decide if we want to undertake that training opportunity, that we're willing to adhere to the protocols that Aimee Neeley and her team and others are putting in place. That's important.
Yeah, just to add on real quick, that's something that hasn't come up really yet, is standardization across different labs. Like, we want to get to a point where any lab can do the DNA extraction, or any lab can do the flow cytometry, but then we need to really have standards, internal standards, standard protocols, and make sure we can trust each other's data at the end of the day, and that, or at least that it's interoperable, and or like if they're all wrong, it's all wrong in the same direction. But yeah, exactly.
I think Emily in the back has had a question for a while now.
Hi, Emily Osborne from NOAA. Thanks so much for such an exciting session. I think the idea of adding biology to GoShip is brilliant. It's very much like BGC Argo, building off the backbone of Argo. We hear in the BGC community that we still are not cycling our floats fast enough to get at biology, and the point has been made that biology is hard. So how suitable do you all feel, and maybe we don't have an answer yet, that the GoShip kind of model, which has been successful for chemistry and physics, is for biology in some of these like worst case scenarios where you might only have a transect in a basin once a decade? How attributable is that change to long-term climate?
Very good question. Especially even with these ghost ship cruises, they're not always done at the same time of year. So there's a whole seasonality component too, which we know biology has a huge seasonality depending on the region that you're in. So whether we're looking at large changes, you know, overall like taxonomic shifts, for example, but again, how would we attribute that? But that's where we have, I would say, like these pinpoint or these single isolated sites that are continuous more often that could help intercalibrate that change, what we're seeing on these once a decade potentially cruises. So, we have things like the MBCO, and they have the LTER that's going out even on those seasonal cycles, so we can start to see how those are changing as well, where they're doing very similar measurements. And then, as I said, we have Aloha and VATS, and there's some similar measurements that are being collected at those locations as well, representative of different oceans. But yeah, it's a very good question, and it's really been challenging. And how will it look in the decade from now, right? So, but we don't know that yet, except from single point sites.
Can I add to that? Is that okay? It's okay right now to say that you don't know the answer to the question, and you don't know what technology you need, and you don't know how you need to grid your models, et cetera, right? But I think there is an opportunity, and again, you know, somewhat shameless plug for two things. One is the upcoming Ocean Sciences meeting, where this is the kind of topic. What does that observing system look like? How do we tackle those biological, ecological problems at multiple scales? Again, back to the grand challenges. That's a town hall topic where you could sit down and talk about that and start to think about what you want to request to the agencies for that kind of support. Where is there NOP opportunities? I think the other piece is the NSF Decadal Survey in Ocean Sciences is going on, and there's going to be a moment very soon where you're going to have the opportunity to submit input into ideas that you'd like to see developed over time, whether that's tech development, whether that's you know, observations on autonomous ships, whatever it is. And so you have an opportunity to think about that as a community and actually provide that input, and that's a strong position for OCB to be in.
We have some questions in the middle of the room.
Hi, Matthias Gabe from the Environmental Defense Fund. I wanted to link back to the CDR discussion, the carbon dioxide removal discussion we had yesterday, because we'll be tasked within that umbrella, as we heard yesterday, of measuring ocean biology, whether it's carbon, phytoplankton, or fluxes in the ocean at a scale ranging from meters to hundreds of kilometers, timescales ranging from hours to decades. And my question is, what does tomorrow's ocean observing network look like to work in service of CDR? What are the big challenges, and what are potential ways to overcome those challenges in the coming years and decades?
I think Dennis McGillicuddy answered that question a couple of days ago. Is he here? I don't know. But he had a view of, because if we use these sustained observatories, and like bats was the example, and if we do some pilots around there, it kind of gives us the ability to capture what we know is the normal. And then we've got that high resolution time series backdrop to say, okay, we've manipulated it, what's the change? But the problem is the scale, as you quite rightly pointed out, and he was basically saying, what was it, 400 sediment traps? Like, you know, let's really make this area the area to kit out and try to do it in collaboration with the people who have been doing those sustained measurements in the past. But I think where you end up with a problem is water moves. So, you know, following that, you could do it maybe in the surface area, but if you're trying to get an export flux, it's a very complicated issue, isn't it? I'm sure other people have.
I think one of the big challenges to accomplishing that is that if you look at it, most of the OCB talks will have a plot showing an anomaly based on a long-term average, which is basically a baseline for either for nutrients or carbon flux, etc. We don't have that for biology, and so it is critical to rely on these observatories that have been collecting biological measurements for a long time to really be able to construct these baselines across trophic levels. So when these interventions occur, we can actually see what the impact on the ecosystems are going to be.
Okay, I think we have time for 2 more questions. Okay, we got one in the middle and we got one right there. So,
well, my question is 2 questions and a comment. The comment would just be that Kevin Becker, who was a postdoc with Ben Van Moy, had this really awesome paper in ISME a few years ago where they showed that chlorophyll peaks at dawn within a Lagrangian setting and then decreases over the day. And so I think that it will be really interesting to see this validation of PACE or PACE with these GO ships or any other in situ validation. And then I had a quick question for Paula and then a quick question for Nicole. The blue Lidar is really interesting, especially when you think about like bloom chasing, or if you're interested in, you have a cruise leaving and there's clouds and you want to know, you know, are there phytoplankton there? And I was curious about if you have any idea like the timeline for when something like that might come online. And then for Nicole, you kind of mentioned this seasonality being really important and these really, you know, intense sampling schemes with these GO ships. Is there any interest in going through, you know, during an El Nino or a La Nina year, or trying to do a summer transect and a spring or fall transect?
Yeah, thanks for the question. So for a blue Lidar, there was one mission that was referenced, Caligula, that's Italian. The Italian Space Agency wants a NASA partnership, is my understanding. It's for a ridiculously small amount of money in the satellite remote sensing world, 20, 25 million, something like that, I understand, with a launch readiness date of 2029. And that's really soon in satellite world, right? It takes a long time to do those things. I think the other thing underway is NASA investing some in the technology to make it more robust for a blue Lidar, to maybe put it on a small sat or CubeSat, because they can actually take active sensors now. And as you rightly point out, there's no problem with clouds, and it goes deeper than passive radiometry. It's, you know, believable to something like 3 optical depths. So there's a lot of advantage there, from everything from ecological monitoring and measurements and exploring to CDR, marine CDR, right? Looking at particle flux and things like that.
Yeah, and regarding the seasonality question, with regard to the GO ship program, that's a sort of a static setup that's done, and so we're just taking it's like a ship of opportunity in a sense to add that biological. But it's a very good question, and there have been studies. So like there's the Gulf of Maine time series, which has been done by Barney Balch's group since the last 20 years, where they're going out seasonality. So there are data sets that represent and actually have, you know, validated a lot of the satellite data that's out there. And then with regard to, like, for example, we have the fairly new Northeast Northeast LTER, which is also going out seasonally. So that's where you can have these regional, it would be interesting to do that in multiple oceans, obviously, to get that variability. But with regard to the Go Ship program, we're pretty much using that as a ship of opportunity at this point.
I think we have one last question. Make it a good one, no pressure.
I'm totally stressed. and worse, you know, if you have a hammer, everything looks like a nail. And I am a modeler, but we haven't heard that in this discussion, I think. And that's one of the key elements of integrating these kinds of static lines. Maybe they're at the wrong season, maybe at the wrong time. Really need to be working together. And I think Paula made this great analogy with the way that the physical oceanographic and the atmospheric communities, their data go onto the GTS, they're assimilated into the models, and we get a forecast of our thunderstorm. And, you know, we're like kind of lagging behind that in the BGC world of modeling, but that's what's going to put these sections into context. That's what's going to help us to know whether those anomalies are due to timing or due to an actual secular change. And so since nobody else brought it up, I'm just going to really say, keep talking to your friendly modeler, and also keep talking about this data problem, because your friendly modeler really can't click on 37,000 locations to get the data that they need for their global model.
I think that's a very excellent comment. Any final comments from the panelists?
Help your modeler friend by, you know, investing time in aligning your data to standards, because anyone that is dealing with large data sets know that most of the effort is going to go toward wrangling those data sets and making sure that they make sense, and then you can filter them and by space, time, taxonomy, etc. So, yeah, it's a valuable investment.
I just want to add something. I appreciate that comment, Victoria. Be kind to each other in the peer review, constructive but supportive, okay? Be rigorous, but understand that there was a time, probably 20, 25 years ago, that I remember clearly, where we were not afraid to put out, you know, data that could be wrong, predictions that could be wrong, and bold conclusions, right? And we've gotten really conservative, in my view, in what we're willing to do, because we're pretty tough on each other, and that's okay. But just remember, it's respect that it's okay to do that, right? Allow yourself that space. I'm giving you permission, and then be constructive and supportive of each other when you review papers like that, because that is how we make progress. And I would argue, again, biology is hard, biogeochemistry is hard, so put it out there, right? We all are interested, and it's going to push the field forward.
I think that's a great wrap up. Thank you so much. Let's thank all of our speakers.